Otherwise, I'll just spend time talking in inaugurations. But anyway, I'm really pleased that you are all here, and I'm sure this will be a wonderful conference. Just a couple of quick things on a personal note. I work with Karthik, I work with Himanshu. We have joint papers in similar areas, and I would have really liked to be here more as a technical presenter than as the dean, but hopefully it'll happen sometime soon. The other thing is my son is actually doing a PhD in biology, and he's very interested only in wet lab biology. So I was trying, I am a chemical engineer, but most of my life I worked in data science, machine learning, AI, and so on. So I was trying to convince him to learn some computational work, learn some machine learning algorithms and data science. And he was completely resisting. He said, no way will I do this. I am wet biology, only biology. That's where the greatest things are done. I said, I'm not going to debate that because I don't know enough, but it's a good skill to pick up. So he goes, starts a PhD in August. And his first rotation, and I knew this rotation business only from sciences. We don't have this in engineering, where students go to multiple labs and spend some time. His first rotation, he's working on, so he went and the professor said, I'll take you for rotation, but you have to work on this project. And the project is actually representing very complicated cell structures in 3 dimensions. And all of that is math and computation. So he called me and then told me, Dad, I've got this paper with use of spherical harmonics and spherical harmonic analysis for characterizing cell structures and so on. I said, sometimes dads are right. Not always, but sometimes dads do say the right things. Anyway, he's been coding and reading math. And I think the relevance of computational work and math for biology has never been more important than it is right now. And conferences like this will really go make an impact in this field. So I'm really happy and thrilled that you're all here, and I hope you have a great conference. And welcome to IIT Madras again. Thank you.
I thank both the deans for that exciting words. So now I would like to request Dr. Karthik and Dr. Venkat to gift Dr. Mahesh and Dr. Raghu with a small token of appreciation. I request the speakers to join the audience. Yeah, so for the first session, it is on space microbiome, and I would like to welcome Dr. Kasturi Venkateswaran, who is the senior research scientist at NASA, the Jet Propulsion Laboratory. His 47 years of research encompasses marine, food, and environmental microbiology. He leads ISS microbial observatory projects to measure microorganisms associated with US nodes, as well as Kibo Japanese experimental modules. He has applied his research in molecular microbial analysis to better understand the ecological aspects of microbes while conducting field studies in several extreme environments. The bioinformatics databases generated by Dr. Venkateswaran's team are extremely useful in the development of biosensors. He has discovered the new bacteria on the filters of International Space Station and named it Solibacillus calami to honor our late president's Dr. APJ Abdul Kalam, who was also a renowned aerospace scientist. I would, before I hand over the session to Dr. Venkat, I would like to request you all to make note of the Slido code and the QR code here. You can scan it and use it to post your question and answers for each session. So I'm glad to welcome Dr. Venkat to take over the session. Thank you.
Oh, I'll say a few lines. Thank you very much for the introduction. Yeah, so it is not going to be interactive discussion here, but at the same time, the Slido will help us to be interactive without interrupting the speaker. So we encourage the speakers to, finish their talk 25 minutes, so that we'll have 5 minutes of, question and answer, which specifically I'm requesting the girls who are doing science here. So I want them to ask more questions, and also like students come forward to put their questions, as much as they can, instead of asking, instead of getting the questions directly from the faculty. Okay, so faculty can find us, but the students and others will have a less chance. So this is your chance to ask any question. None of the question is a stupid question, so don't worry about that. Ask your question, but if you think that you will be asking those questions at the end of the session, you will be forgetting what you want to do ask. So then and there, just put your questions in the Slido. Is there any question on the Slido here? Anybody have any issues on putting your stuff on Slido? That's the only way that they'll ask the questions, right, Karthik? The Slido is the only way to ask the question. And it is there for the session chair to see and frame the question, right? Because we are not requesting the person to ask directly the question. Only we are reading your questions. If similar questions are there, we will put all those things together and do it. Okay? Yeah. Okay. Do you want to say something about the Slido? Is everything good? Are you sure? So you are going to give something to the session chair so that they read, or how do they get those things? But what if the questions goes beyond that slide, one slide? Okay, but okay. Okay. Let me get first speakers out first. Again, sit there. Okay. Our first speaker is Dr. Nick Bernardini. There may be a spelling mistake here. His full name is Benar. Okay, Bernardini. He's our planetary protection officer. So what that means is his signature is fifth in line from the president for a life detection mission that is flying. He's our planetary protection group head. He's sitting in head office in the Office of Safety and Mission Assurance. So he has conducted research in the field of environmental microbiology, studying microbes in extreme environments. He has participated in both applied research and field studies in conducting molecular and traditional microbiological community analysis. He has assumed various roles in the past, such as leading the sampling and laboratory support team for the Mars Science Laboratory. And he has served as the lead for the InSight as well as March 2020 mission. And he was at JPL, before heading into the NASA headquarters for more than 20 plus years. And he's going to tell about that also. And for the students, he's a very inspiring person because he started the career as a student intern in JPL, and now he's heading the whole, planetary protection office at headquarters. Here is, Nick. Presentation. Presentation mode.
Full screen mode.
Huh? Which one?
Full screen mode?
Yeah. See?
Awesome. Yeah. Okay. Thank you, Venkat, for that warm welcome. And thank you, Karthik and the Institute for your hosting and having us here to have these conversations. So looking forward to the next couple of days of conversations. So jumping right into space biology topic, planetary protection. So this is kind of overarching for a lot of our missions that we have as we go to explore. And then certainly as we're thinking about bringing samples back. Cancel. So what is planetary protection? So a lot of people confuse planetary protection with planetary defense. So planetary defense, we had a recent mission, the DART mission. This is a mission that went and directly impacted an asteroid. This is our planetary defense office. So that deals with large scale objects that are hard, that you need a telescope to work with, versus planetary protection deals with small scale, squishy type of biology. And certainly we need microscopes to work with. So vastly different scales on the order of magnitude between planetary defense and planetary protection. So we think of planetary protection as a way of maintaining safe and a sustainable exploration of this solar system, particularly in terms of biology. So when we go for forward planetary protection, quite simply, how can you look for life on other planets when you bring your own? So we have a means of policy and infrastructure that we look to implement on our spacecraft to solar system target bodies such as Mars, Enceladus, Europa, that have high probability of life. So we don't want to bring our own life there and contaminate it. Likewise, when we bring samples back from those target bodies, we want to make sure that we don't cause the next COVID, right? So we want to ensure that we're not providing any harm to Earth's biosphere. So we have that balance of forward planetary protection as well as back planetary protection. So we do this throughout the mission life cycle. So we start with mission design. So this really helps define our risk classification. So where are we going? Where are we going to explore? What are we doing in that mission? Are we just drilling on the top 10 centimeters of Mars? Are we just looking for science, or are we bringing things back? And then that impacts our mission requirements, and then what we do ultimately for the mission. So for example, if we have an Earth-based orbiter going in low Earth orbit, there's no planetary protection. So it's simple ISO class clean room that's based on your satellite needs, versus if you're going to bring the rover to Mars, that's when we start to think about the levels and layers of biological contamination cleanliness that we would need for our spacecraft. So throughout the mission build, we go through extensive measures within clean room, dressing in full bunny suits to ensure that we don't contaminate the spacecraft. We're constantly cleaning with isopropyl alcohol, dry heat microbial reduction, some sterilization, vapor hydrogen peroxide to lower the limits of Earth biology onto our spacecraft, preventing recontamination, and then ultimately launching a clean spacecraft through to get to the surface of Mars, for example, here with the Mars 2020 launch. So we have to think about biological cleanliness in a holistic approach throughout the design of the mission. Really starts with the types of materials that you're thinking about. So if we have high heat sterilization, We have to be concerned about optics or various engineering components, electronics, whereas it's detailed for early on materials and process considerations for the mission. And then all the way up until launch. So once we get to our launch vehicle, there's certainly a large environment that we have to be thinking about interfacing with throughout the whole launch environment, and how that interacts with the spacecraft. So we can't go through 5-6 years of clean spacecraft build to simply put into a dirty launch environment. So we have to be thinking about all the steps along the way as we're building up the spacecraft. And then we have a lot of operational considerations too, for once we actually get to the surface of the environment, planets, as well as if we have an orbiter, what do we do with that orbiter at the end? So we can't just quite simply smack it into the Martian surface. We have to be thinking about maintaining it into safe, stable orbits, or ensuring that it has multiple years of consideration for seeing various sterilization, think Martian UV, for example, before it deorbits into the Martian surface. So a lot of our requirements right now are very prescriptive requirements. So these are our spore-based requirements, bacterial endospore. So we have 5 times 10 to the 5th spores on our total spacecraft at launch, with surface area of 300 spore per meter squared. These are our common international guidelines that we all work towards. And we also do a lot of reporting out. So we think about the types of organics that we're sending, because obviously they'll impact a lot of our mass spec and other science endeavors, as well as keeping track of the bioburden on the actual spacecraft. And so we go through a series of cleaning. In the middle, this is one of the examples of what we would do to verify the spacecraft. So we think of the spacecraft as like an onion. So as the spacecraft gets built up, there's various parts that get closed out. And so we have to go and clean and sample to verify what the biological load is on those particular surfaces as the spacecraft gets built up. Clean. This is probably the hardest job. It's easy to keep something and get something clean and deliver it to our clean rooms because it's very well small teams, small clean rooms. But once we get into large integration facilities, this creates quite a bit of problem because we have multiple teams coming together, very large clean rooms, very large ground support equipment, and these are high potential for contamination. So we have to balance that throughout the spacecraft build. So as I mentioned, the bacterial spore is kind of our gold standard. We've done this since the 1960s, traditional microbiology, but obviously we should modernize. We have a lot of modern techniques, and this is where a lot of the omics come into play, and really kind of unlocking that potential, as I alluded to with the title of this talk. And so this potential allows us to have a more technical conversation, get out of the prescriptive state, and certainly think about the potential of other life other than spores as an indicator for Mars and potentially even for outer planets. So here we are going to Europa coming up in 2024 with the Europa Clipper mission, and certainly thinking about other outer planets missions. So what's the appropriate biological indicator for these planets? Is it the spore like we have for Mars? Perhaps not. And so that's part of the conversation that the omics world allows us to have. And so while we still have these as the gold standard, there is no international standard for this. And so it's only very agencies prescriptive. So NASA, as well as the European Space Agency, has it well documented. And it's everything you would envision with traditional microbiology, with swabbing, and wiping the spacecraft surfaces, to doing extractions, petri dish plating, and think hundreds of thousands of petri dishes throughout the course of a mission. And what kind of data is that providing us? To what potential can we be getting with other omics data? So far to date, from a Mars perspective, this has treated us fairly well. It's been a balanced approach. It's allowed us to keep our missions clean with something that we know how to do, that majority of partners that we work with can accommodate. It's very easy entry, low level, but that's worked out very, very well for the type of exploration we've done. We've essentially gone to areas of Mars and access areas of Mars that have a lower likelihood or potential for life. But as we're starting to increase our capabilities from a scientific perspective, limits of payloads, getting more sensitive, as well as wanting to go explore polar regions or special regions where we could have ice present, where organisms may be present. We have to think about, what does that mean for the cleanliness of our spacecraft? Is the spore metric still good enough? And so this kind of allows us to be thinking and have conversations out of that. And just a little bit of history behind that, though, is that a lot of these thresholds were really fundamentally oriented around the 1960s, when it was United States and USSR at the time. And so the risk posture for thinking about biological cleanliness and exploration of Mars was really just in that context of what missions were going to be done, and what were the international players look like. So obviously we know that's changed quite a bit within mission cadence, as well as the players involved, commercial space, other spacefaring nations coming up. So is that posture still the same? So these are all kind of thoughts that we're starting to really have conversations about at the international level of how good is good enough? Are we there? Is SPORE still the right level? And eventually, once we get crewed mission to Mars, certainly we know that when you bring a fermenter somewhere, the human, biological cleanliness has a completely different thought. So that spurs different conversations, and that's kind of the forward thinking that we're leaning towards right now with policy and our verification approach. And certainly it's just not about Mars. As I mentioned, Europa, outer planets. There are other planets that have potential for life, and we want to go explore those and be able to land on those planets, do close flybys like we did just this past week with the Juno spacecraft. And certainly there's a whole nother wealth of galaxies out there that potentially have life as well. So we want to make sure that we're having a sustainable exploration for that. And so again, a lot of questions, but what about those cleanliness on the target bodies? What types of organisms should we be thinking about and considering? And our omics and other approaches allow us to have that technical conversation as opposed to using strict indicator prescriptive, bacterial spore approach. And then certainly once we get life detection missions back, we're not going to be culturing. The plan is not necessarily go to a culture-based technique to assess what's there. So we want to be using the most state-of-the-art samples, the most state-of-the-art approaches when we're bringing samples back sometime around 2033 is when our current plans are for Mars sample return. So the prescriptive requirements really allow us to kind of specify the what to do it, how to do it, but really thinking about larger, we're wanting to kind of adapt ourselves into a more performance-based approach that allows for assurance case conversations and development, other kind of high consequence, low risk environments such as nuclear industry are using, oil and gas industry is using. And so we want to be able to modernize the way we're thinking. So kind of getting into a not how to do it prescriptive approach, but what to do, and really getting to that balance. And it allows for those conversations to have that flexibility, to have those reduction in thought conversations as we were introduced this morning, and to really explore the trade space with current science as well as engineering inputs. A lot of people think, oh, well, assurance case, performance-based requirement sounds great, but that's just simplifying things. It's not relaxing the requirements we have. It's actually harder to prove because you have to have more people in the room. You have to have a larger international conversation. You have to have a larger peer-to-peer conversation within the scientific community. And that doesn't necessarily allow for a get-out-of-jail-free card here. So I think that's the big concern that most people are having, but it's actually harder to prove this case because you're building it from the ground up and tailoring that for your specific missions. So some things to think about as we're kind of at this crossroads. Is it appropriate? Does the case that you have, the technology that you have today, is it mature enough for what you want to do? Is there enough scientific consensus that would allow you to leverage omics approaches for planetary protection and spacecraft? Is that risk posture good enough? Do we have, when you're going through the bioinformatics and seeing unclassified, unknown percents, is that acceptable for assessing risk and whether life is going to be able to be propagated on other planets? So these are all questions we have to ask ourselves, both from a scientific perspective and then the responsive side from a policy enabling perspective. But it presents an interesting problem, at least omics does, or any biology problem, because from planetary protection, we have a large range of environments that we're essentially wanting to work towards, as well as biomass. So we think of the ground-based robotics, very low biomass work. This is where we've built a lot of our Mars rovers, but certainly going ahead as we get into lunar exploration, Mars exploration, planetary protection starts to really become involved and on orbit. So we have to be able to have microgravity assessments. We also have to have reduced gravity once we get to Mars. What does it look like for the habitats we're building? How do we monitor that? Eventually, when we get Mars samples back, we're going to want to be able to assess whether or not there's life. And then to ensure that we're not getting into any back planetary protection issues with crewed health, there's an interplay there as well. So we have to be able to balance the human microbiome with what's happening with the external environment and parsing that from crew health to what the environment is causing. So there's a large range of potential trade space that we're looking at and kind of evolving out of, whereas right now we're just more or less focused on the ground-based robotics stage. So how do we do that? So we've got a pretty complex roadmap that we've integrated throughout NASA for planetary protection. Won't necessarily take you through all of that, but there's a whole component here at the bottom just to bring your attention to, where it's looking at the microbial monitoring output. So we have an existing ISS that Venkat and others will be talking to you more in depth about, where this is a constant monitoring of the built environment. When that's being applied to and really looking at our lunar and Mars exploration coming up. And so we have a whole integrative plan throughout the next decade to really focus on the ability to leverage existing missions, both on the ground to build up the technology, low Earth orbit, and then eventually Mars to kind of realize the technology for planetary protection and microbial output. So it's looking at that synergy of what we have existing on microbial monitoring and what could be applied for planetary protection discipline. So one of the ways we do this is scientific consensus and having conversations like this throughout the workshop. So throughout COVID, we had a nice workshop chaired by Stephen Green, who's going to be talking to you later in this talk. So we wanted to assess from a NASA perspective where we're at. Are we there? Does the scientific community think that we're able to start to adapt omics into our planetary protection use case? So we hosted a one-day workshop with participants across NASA, academia, and the US government to basically look at, are we there yet? So in doing so, the answer was yes, there's an opportunity to do that. But there's a lot of key knowledge areas that we have, particularly in sample collection, sample processing, and sequencing with like kitomes, for example, and background noise with low biomass considerations, with bioinformatic analysis. And the other part of this is the whole quantitative risk assessment. What organisms do we care about, and how do we apply that to our specific target body and spacecraft use? So I think one of the key takeaways here is that, not only is a metagenomic approach good for both phylogenetic and functional characteristics, but there's certainly a component for quantitative analysis using digital PCR. And so we can kind of use these both together in a coupled vision to be able to modernize our spore-based assay. And we're looking at starting to implement some of those things now. So in doing that, one of the jobs that I have at NASA headquarters is looking at the pathways we have to enable that, to align our funding in all of these strategic areas, to work with researchers and technologists to infuse those in the missions, and to also have this biological risk posture conversation and safety posture, because certainly we can have a balance of mission success and mission safety, and we have to have that balance. You cannot simply just have one over the other. And so are we there yet with really getting, as this technology is advancing, how does that adapt our thinking with this risk posture and safety? Coordinating, obviously, within the Committee of Space Research and other international forum for being open and transparent with the processes that we have, and leveraging really the scientific peer-reviewed process is one main contributor in doing so. And then certainly we're starting to continue to develop a lot of our policy with drafting operating protocols for genomic processing. So with that, this is it. So thank you.
Hello. Okay. Thank you so much for a informative talk here from the Planetary Protection Officer. And there are a lot of questions and around here, and it's really a very good way of interacting people. If you see on the right side, there are popular thing is coming. So let's go with the popular
Top one.
thing. The popular thing always pop up at the top.
All right.
So why don't you answer the very first one? We
Sure.
see strict guidelines for forward contamination, but we're also talking about human exploration and settlement. How can both be simultaneously done?
Excellent question. And this is what keeps us up at night. To be honest, this is really the work that we have ahead of us is really embodied in this question. As I mentioned before, once we send humans, we have fermenters sitting on Mars. So part of the understanding is several-fold. One now is we are in robotic exploration, and so we want to make sure that we're making and having capabilities to get the most pristine samples for science before the humans get there. So that's one component of really why we're trying to push hard for Mars sample return right now. The second thing is we have had a series of international conversations for establishing knowledge gaps for crewed missions to Mars. A lot of this looks at and recognizes the fact that we as humans are going to go, and there's going to be communication of human microbes to the Martian surface. And likewise, when we go out and explore, the Martian environment is going to be coming inside to our environment. So with that, how do we really think about a safe and a sustainable exploration strategy? So one example of this is kind of the way that we have national parks and our nature preserves, in the sense that, you could send the human to the known area, and then if you want to do more detailed exploration, this is where the robotic and human interface could lie. And so then the robot can go that's more clean to those pristine areas to bring the samples back for our scientists to do further analysis on. So.
Thank you. There may be some follow-on questions from there, but we will see how popular that is later. But this is a very interesting question. So I think for sure. Won't some extremophiles survive some of the harshest clean treatments sometimes? What do you do in this?
That's a good one as well. So this is one of the problems of if you're just using the spore as your bioindicator, what are you missing? So I'm a huge proponent of looking at omics and kind of a fuller-based technology to really understand what's there. And so certainly you're going to be missing a lot, and you're not going to be able to kill everything with every given heat, microbial reduction technology, vapor hydrogen peroxide, et cetera. But the reality is that our spacecraft aren't clean. Like, we have to recognize that there's a risk of biological contamination for every activity we do. And this is the mission success versus safety balance. And so we can't simply say that everything has to be sterile. Otherwise, we can't meet mission success. Like we can't fly a mission because we can't have optics. We can't have electronics that support these types of harsh radiation and other biocidal effects. So you have to balance that safety side with having some organisms and potential with that.
Yeah, so one other thing for the same question is, cleaner is not measure, obviously. Are we actually making superbug by cleaning too much?
So that's a good question. You could probably answer that better than I. Honestly.
Yes and no.
Somebody else talking.
Nitin maybe wanted to answer. No, I think very much we can, potentially we can. I think this is, what we see in medical industry too, right? Of making sure that we're swapping and having the right biocidal changes out of our clean room and the way that we clean, and not just pushing one type of cleaning all the time. So I think, we have to consider all of this hardy organisms and the way that we think about biological indicators and we prep our spacecraft cleaning. So.
That's right. Yeah, so, there may be more discussion that we can do there, but we'll go later. Now the other question, because I'm going with my time, is right now is 9:55, not that time. Okay, so the other question is the top one. Go ahead. When bringing samples from Mars, do you mimic the Mars condition for the microbes to be alive such that it can be analyzed? How unique are those conditions?
Good question. So as of right now, we haven't brought samples back from Mars. So I just want to clarify that because everybody's misconception is we already have samples sitting back in a lab. So this is what we're currently doing right now on the rover that's roving around Mars 2020 Perseverance on Mars, collecting cores. So we probably have, I think, 10 or 12 bits or cores that we've collected from the Martian surface already. Actually, Ganesh is one of the key players who was sampling the spacecraft during its build here. So you can talk to him more about that and how that process went as kind of a local source, if you will. And so, so basically, there's a lot of science requirements that go behind that. International science community has set requirements on temperature, as well as various sealing requirements for the tubes and the various vessels. But once they come back to Earth, they're going to basically go into an astromaterials curation facility, going under inert gas, argon, nitrogen, for example, and kept at ambient room temperatures. And so that's how they'll be received when we get them on the back end. And I think part of this is really, how do you think about life and what we're looking for life in those samples? There's actually a whole framework that we're putting in place right now of trying to understand how do you look for life, unknown life, when you don't know what you're looking for. So it's kind of looking at key fundamental things of life, of physical structure. And so there's a whole microscopy component that you would look. Does it look like life? Do I see cellular structures that I recognize? Are there biotic, organic carbon compounds that may be present? And really leveraging a multitude of diagnostic technologies, but not necessarily culture-based, to assess whether there's life on Mars in those samples.
Great. So some people are asking about how to ask the question. See, for those who cannot, not started this from the beginning, there is a QR code there. Just scan and then type whatever you want. Okay, that's good. Now let's do the other popular question that may be the last, I think, because it is 9:58. So if a human mission is sent to Mars, will spacecraft cleanliness still be as critical? How will human presence on Mars affect future life detection?
So the answer is no. The spacecraft is not going to be as clean, at least from a human built environment perspective. That's given and recognized within the community. There's actually guidelines, Committee of Space Research has put together guidelines saying that we're not going to have it as clean as the robotic spacecraft. It's virtually impossible. And this is really the interplay of capturing the types of knowledge that we have, what types of organisms that we're sending and monitoring that, and the potential release of the human impact zone, if you will, and having that balance of going to known regions in Mars versus potential robotic exploration. Thank you.
Thank you so much. Can you close it? Well, close it, yeah. Thank you. So, wonderful talk. And there are so many other questions that will be answered, just send a, we'll try to answer those things, if it is available for us. But in the interest of time, we have to move to the next speaker, which is virtual. Please get the virtual things ready while I'm introducing the speaker. Dr. Nitin Singh, he is going to talk on the plant growth in space and microbiome. So, he may be talking about when he joined to JPL, but he was our postdoc to join about a few years, 5, 6, 7 years back. And then now he's a JPL scientist and having his own tasks to lead. So he has 15 plus years of research experience in microbiology, studying microbial taxonomy, genomics, metagenomics, and microbial ecology study, and also its interpretation. He is specialized in working on pathogenic microbes found in clean rooms and hospitals to understand how microbes adapt to become more pathogenic under anthropogenic pressure. Right now, in addition to what we're doing in JPL on various missions, the metagenomic analysis of various missions such as, ISS, IMPAS, SpaceX, JPL, SAF, Mars 2020, etc. He is also serving as a permanent member of Caltech Institutional Review Board, Institutional Animal Care and Use Committee, ICAC, and Institutional Biosafety Committee. Here is Dr. Nitin. Is he there? Nitin, are you online?
Very much.
Okay.
Hey, good morning, everybody. Can everybody hear me? Can everybody see the screen? I just because it's too much loud and clear. Dr. Venkat, can you confirm?
Yes.
Okay, thank you. Thank you very much. I'm really, really excited to be part of this. So very good morning to you. It's been night here. So yeah, happy to be on the other part of the world and speaking to people on the other part of the world. So very good morning to all of you. I am Nitin Kumar Singh. I am a scientist at Biotechnology and Planetary Protection Group at Jet Propulsion Lab. And I will, as Dr. Venkat told, I will be talking about plant growth in space and microbiomes. Moving ahead, a brief introduction about myself. And as I'm talking to young people, maybe they can take something out of it. So I started as a plant biotechnologist. So that was my whole starting when in 2003 and 2004, when I started, I was a plant person. You can see like I worked on Vitex nigundo, and this is the actual field that I was guarding so that the animal doesn't eat my experiment. So that was how I started in science. And then I moved to animals during my master's. During my PhD, I became a microbiologist. I did my PhD in microbiology. And since 2016, it has been 2,455 days. I'm being in JPL. As Dr. Venkat said, I'm a scientist here. I work on different missions like ISS, Mars 2020, and spacecraft assembly facilities. I'm also IACUC, IRB, and IBSE member at Caltech. That's the observation. And when I'm not doing science, I'm really talking to people. It's a pleasure talking to you today. And if you feel like this face is known, maybe because you saw this interview that I gave, after which I got to know how much people can love you and hate you at the same time. And when I'm not doing that too, I'm reading books. I like reading about philosophy and technology. So, and sometimes I do use my microbial taxonomic abilities to name humans. And this is my niece, very happy to be named by a taxonomist. So the idea to tell you about me was, if things didn't went according to your plan, doesn't matter, because I am one of those person who was not able to make to IIT or a medical institution after 12th. So there are better plans. Moving ahead with our topic today, plant growth in space and microbiome. So here is a brief outline. So first we will talk about why do we need to grow plants in space, challenges, how have we done till far? It's a sort of a history that we will be talking about. Then ISS and plant omics in International Space Station, what major findings we have, what curious cases we have, and then we will talk about how JPL comes into the picture, because JPL is a robotic institution. What is JPL doing in this whole plant scenario? So why do we need to grow plants in space? That's the elephant in the room question. So with our ISS resupply mission, we know that a normal adult person requires 8,000 pounds of resources of oxygen, food, and water every year. We have technically grounded it to that level. Now, for long-term deep space exploration, we need a long-term life support system, like the Artemis that we will be going to have in 2 years, where we are planning to put a station up there. We need a long-term support system and technologies that can actually be supporting food and production of food and breathable air. And also the idea of going and studying plant in space is how living organisms behave towards space radiation, extreme temperature, and lack of habitable atmosphere. That's the major question. We need to grow plants in space. One of the other questions that is always asked to me when I'm talking to people is, why do we invest so much in space, plant research, or space research together? I would suggest you to please go and read this International Space Station Benefits to Humanity. It's a very good read, and it tells you about the amazing technology that came out of International Space Station and is being used by humanity. Your mobile phone or the smartphone looks the way it is right now is because a lot of technology that was developed in the International Space Station has been implemented there, especially the selfie camera. Getting back to our topic, so the prime requirement for deep space exploration or human establishment on Moon and Mars, we will take this term together, requires atmosphere regeneration, water purification, waste processing, and food production. Beyond the physical technologies, we have to design something called a life support system, which will be a designed symbiosis between crew and other organisms, namely the higher plants and the microbes. Now, when I say designed symbiosis, it will be forced. Because if we go on Moon, there is no microbial cycle or neutron cycling happening there. So this is something that we take for granted on Earth. There will be no decomposition of the waste that you throw. So you have to force that decomposition there. And a thing I mentioned here is a Mount Everest dilemma, is because imagine the people and the climbers going to Mount Everest and they are dumping so many things out there. It has become the largest dump site or the highest dump site in the world. So what we are trying to design here is an output research station rather than an extraterrestrial dumpster. So we have to keep that in mind while designing our deep space exploration, whether it's Moon or Mars. Currently on ISS, approximately 90% of the water, including the urine, and 40% of oxygen consumed is recycled using a device called ECLSS, that's Environmental Control and Life Support System. You can see a mock-up of the photograph that's provided at the right side of the screen. So that's reason, and for everything else, they are dependent upon the resupply mission that goes every couple of months from Earth to the International Space Station. Talking about the challenges, so challenges of plant growth in space environment. So the biggest challenge is gravitopic set point angle. Now, the gravitopic set point angle is what guides how the roots and the shoots will be formed, because the roots have to go down and shoots have to come up in a particular direction where the sunlight is like ample. So that is like decided by the gravitopic set of angle. And now when this whole microgravity comes into picture, this gravitopic set point angle is disturbed and the plant has to adapt. And we have seen that not all the plant adapt equally to this thing, because if we see the Earth, if Earth is 1G, Moon is 1.6 and Mars is 0.38 G. So this GSA will be different for different plants, and maybe it may be lethal for some of the plants. Beyond gravity, we also need to take care of something called space radiation, light, temperature, atmospheric composition, and all the other parameters has to be controlled so that we can have a sustainable growth of plant in space whenever we are going there. Few of the examples that are in test are Eden, like the one shown in the photograph, if you can see the cursor. This is like, it said that it can generate 268 kilograms of food in under like 12.5 square meters of area in 9.5 months, because the rest of the 2 months it's technically like not producing anything or closed or waiting for the resupply to happen. Then there is something called Melissa. This is developed by European Space Agency. It's called Microbial Ecological Life Support System Alternatives. Now, again, wherever you see the elephant, there is a question there, because you can see how much huge these instruments are and how much they are dependent upon the supply from human. Now, imagine this taking these to a different planet or a different satellite will be a huge task, and it will take something like a Starship level of cargo pulling rocket that can take these things. So otherwise, we have to like break these instruments into smaller projections, and we have to make sure that it's sustainable. And when we say sustainable, it should be of autonomous nature, not dependent upon the Earth supply. And still, there's a big question how these like functional instruments will behave when they go on an independent journey to deep space. So how we have done till now. So till now, before preparing for this particular presentation, I didn't knew that we go as back as 1946. I was aware that there were experiments done in 1970, but this actually gave me it was a learning experience for me too. So in 1946, America launched a V2 rocket, which had mage seeds in it. And V2 rocket is actually popular for this first photograph from the space, which was given by the V2 rocket. And you can actually see, like, since 1946, we had a photograph which shows that it's like pretty much circular, because every other day I get like some random message on my social media that Earth is not round. So, and I cannot like, that's the reason of putting this picture here. We are very much aware of that. So in America started the whole plant experiment in 1946. And there was sort of a competition going during that time. So Russia made an effort. So America sent 1 seed, Russia sent 4 in 1960. But in 1966, they did something more ahead, went ahead. So Cosmos 110, actually what it did a germination in space. This was the first germination that was reported in space. They also sent 2 dogs up there, and then they later retrieved it for analysis. In 1971, again, US Apollo mission, this was Apollo 14, they sent the whole 500 seeds around the moon, got back to Earth, and then they started growing them. This was called moon trees. The one tree that I have actually like put the photograph in because it's nearest to my home. It's 5-hour drive from our home, and would visit it sometime. But some of the trees were also planted in Germany and Switzerland, and these were observed for a good 10 years before they were actually left to grow, because they thought that something different will grow, or the tree will grow something differently. But that never happened. Biology doesn't works that way. Then came the US Skylab, in which the actual good radiation rise experiment were done for phototropism in microgravity. Then again, it was because the competition was going on, the Salyut experiment happened, where the first time something called a seed to seed experiment were done. And we came with a concept called plant growth platform, like Oasis, Vazen, and Python. These were the first platform to be implemented in space for plant growth. And then came the era of space shuttle, where for 20 years, we did a good load of plant research on space shuttle. And we first time US started testing the plant growth platform, which were like plant growth facility and plant genetic bioprocessing apparatus. And then the whole scenario changed after the International Space Station came into picture, and the whole area of plant omic research in space just exploded. These are the few examples right now. This is Kate Rubins with the advanced plant habitat. And then this is a veggie experiment currently growing lettuce. To capture the whole what's going on International Space Station, it needs a total different topic, and hence a new slide. So International Space Station gave us an opportunity to study the same experiment in space and same experiment on Earth together. So it was space-based investigation and ground-based investigation of direct and indirect impacts of microgravity and ionization on different stress response, oxygen biology, cell wall biology, plastid biology, and mitochondrial process. And the omics actually exploded because there were so many first that was done on International Space Station, like first transcriptomics, proteomics, metabolomics, lipidomics, genomics, glycomics, and other 53 omic terms that you can actually name off. While I was preparing for this, because I actually started preparing for this particular session a month ago, but this review just came out 2 days ago, the Plant Space Biology in Genomics Age. If you want to start with plant space biology or want to study about it, this is a good 25-page read I would recommend to start with. And then there are because they left a lot of topics also, but that's just to start. If you want, it's a good read. Major findings that came out of the plantomic experiment. Yes, it affects the genomics, transcriptome, and proteome. And the effect will be different on what type of test plant it is, what type of ecotype it is, what type of genotype it is, what type of developmental age it is. There are post-transcriptional modification, and most of these were studied on callus. Callus is like a non-defined where a plant where the organ has not been defined and the rooting and shooting has not happened. It's just a callus mass. So most of the study were done on the callus mass of post-transcriptional modification and epigenetic changes that were observed during the generations. One of the things that was major finding was that the thickening of the cell wall was lower. The rigidity and thickness of cell wall was lower in microgravity, and hence the division was faster because less energy was being spent on division. So the cell proliferation was faster. Arabidopsis became the workhorse plant. Like if you see 10 plant experiment on ISS, 8 of them or maybe 9 of them are done on Arabidopsis. But one of the major findings that came out in Arabidopsis was phototropic response to red light. And the one that actually excites me a lot is the positive phototropic response in the root. And this is you only see in mangroves in journal condition where you have new metaphors that comes out of the soil and further towards the atmosphere to suck in air. And there are so many others which I was not able to capture here because there are more than 200 of them. But these were the major findings, which I'm trying to keep this pure science rather than application. Coming on to the curious case, yes, not only human, but spaceflight condition make plants more susceptible to pathogen colonization. Keep in look for Venkat's presentation because he will be putting a Xenia case. And this, nobody knows the answer why plant behave in this way or why the immune system of plant is weaker, maybe due to radiation. That's one of the intelligent guess that we can make, but it does shows that. We know microgravity, humans are immunocompromised. They are more susceptible to disease, but plants shows the same behavior. One of the molecular hallmarks for the stress response in plant, which we actually observed while we were developing this tool called Mango. So Mango, we did a whole bunch of data processing, and we always saw this heat shock protein coming up time and again in all the data set. And when we rechecked it was reported by so many people. And why it actually surprised us because even though it's a heat shock protein, but the place they were grown in International Space Station is highly controlled, highly temperature controlled. There are like hardly a degree of change while the growth is happening in International Space Station. And we know that HSP is induced by osmotics and salt, UV, pathogen, and wound stress. But we are talking here about healthy plants. And we saw, like in all of the data, the HSP has shot up. And mostly those HSP were 17.6A and HSP 101. So if you really want to check on these data, they are available in our NASA Gene Lab resource. Right now, there are 57 plant-based Gene Lab resource. If you want to check the process data, you can definitely go and check on Mango, and you can find all the plant pathogen we have categorized in, like all the pathogens that we found in the International Space Station, whether it's a human pathogen or a plant pathogen, that has been well documented here. And if you want to start your astrobiology journey with Arabidopsis, the TOAST is a very good database to start from. It's both the tools were made by JPL in collaboration with Wisconsin, University of Wisconsin. So yeah, feel free to check it out. Now coming to JPL, what is JPL doing in this whole plant business? So till 2018, JPL had no plans to go into the plant business. Only it was in JPL where the BISRU, which is Biological In Situ Resource Utilization team, had a meeting in December 2018. This is the first pick of the meeting, and you can see at that time I used to have hair. But the concept came up, and the winner concept was a lunar plant growth chamber. And the mission that was decided, the mission goals were given to us to demonstrate that lunar regolith can be converted into a soil for a plant growth, and prove that lunar regolith does not prevent plant growth, because it should not be lethal to the plant growth. And there were multiple variables that were identified at that time. One was inoculant, which microbes, which we will be talking later, regolith particle size, growth medium, like what concentration and what amount if we ever want to put, and gas mixture and seed variety. So these were the variables identified in that meeting. And the reason of telling you that, like, this is how a idea is born in JPL. This is how we mature it. So like this happened in 2018. Next 1 year was decided in, okay, how this could be a different. And one of the things that why we had to do a plant growth chamber, the reason is, if you see the whole plant growth platforms that has been used since 1970 till to 2020, these are all dependent upon fertilizer. They are not in situ resource utilization. None of these devices were meant to optimize for ISRO lunar regolith use on moon. And advanced plant habitat is right now in International Space Station. Very soon, this may be launched to the International Space Station. And also none of these are closed loop. That means, like, when you are studying the gases and nutrient fluxes or the cycles, it's hard to predict where the things are coming from. So this chart was taken from this particular review, the review and analysis of over 40 years of space plant growth system. And it's a very good review. I would recommend, if you are interested in plant, definitely go and read. But during this time, what also happened, that China's Chang'e 4 lunar lander actually grew a plant on moon. And it was like, it took all the resources from Earth. So it became even more important for us to definitely prove that we can use lunar regolith on moon to generate a plant. So we were working for like another 1 year. We tried to design the design concept was this whole instrument should be able to successfully grow and record data on plant growth in lunar regolith. The ideas that were the challenges that were given to us were it should be upgradable, it should be 3D printable, easy to access, closed loop system, diverse sensor interface, it should be autonomous, accommodate different type of plant growth substrate. So these were like sort of big challenges while we were doing this. It took like a good 1 year to get the whole idea together. But our luck, the whole world went into a shutdown. And this was like March 2020 when we last shut this project. And it was not until March 2022 that we again started it. And this was the first version. Right now we are on the third version but technical documents technical designs are not to be shown in public. So this is what it looks like, the version 1. So this concept was funded in 2022 for the rapid, from 3 months. In 3 months, we were able to make the version 1. Currently, what we are doing with this plant growth chamber is testing and integrating a water sprout, because this has to be totally autonomous. There has to be like minimal or no astronaut time has to be spent on this. And then we are trying to get into a AI and ML software that will manage all the environmental control and collect and download data. And the life, this.
Nitin, you have 1
Yes?
minute. Only 1 more minute.
Okay. So, and okay, I lost my track. Okay, that's fine. I will try to be faster here. So, and we wanted this instrument to be like having a 30-day cycle. So other things, what we did with this, we quickly did a lunar regolith experiment, and you can actually see the plant actually growing in the lunar regolith. This was a mixture of a soil and lunar regolith, and with some like garden soil substrate were used, so that we know that actually it can support. And this is from the actual camera that the instrument has, and the whole, we recorded it for 14 days till it actually like broke, and then we have to make a new one. So once we have the instrument, the next was the having an organism. I will be talking about 2 organisms, because Dr. Venkat will be taking it a more deep dive into the microbiome part of it, but 2 organisms and why we are doing it. So this is Agrobacterium tomcornis. It's known to support plant growth. We were able to identify it from International Space Station and compare it to different like nearby species of genome species 3, and we were able to prove that this is a novel species. One of the interesting thing that we came was this particular part in the genome, which we like don't know what its function is, but it's very much unique to this particular agrobacterium that came from International Space Station. Another organism that we are actually very promising organism that we found in the collection of 6,000 organisms that we have is Sphingomonas, which actually produce auxin-like substance, which promotes plant growth. And this is why we are doing is because we are collecting 1 by 1 growth supporting organism, which we will use as a synthetic microbiome in future when we go to Moon or Mars, and when we want to use lunar regolith or Mars regolith as a growth medium. Future, yes, we want to have adapted seed. This is microgravity adapted seed and microgravity adapted assembled microbiome to be tested on the International Space Station Advanced Plant Habitat. And we are trying to develop our apparatus for Artemis 3 mission, which will be a mission, a manned mission to Moon. Thank you for your time. This is our team at JPL. Anna and Laura are soil scientists. Seth is a cave microbiologist, and Atul is a real one eukaryotic scientist. And you can actually see a very hungry Venkat here. He has been supporting this for long, and Mu, who is our group supervisor at JPL. I know it's too early to say you like enjoy and go party, but whenever this day is done, please enjoy your time. Thank you.
Thank you. Thank you for the wonderful thing. Are you able to read those Slido stuff?
Let me stop sharing and then I can just go.
Okay.
Okay,
so,
let's see what's there.
Wait. So we don't have too much time. So the popular, what wins actually. So the top one had 4 votes so far. So what was the photosynthesis efficiency for a plant on planet Earth and the same plant in the ISS?
Okay, it's almost nearly the same. You have a 10% difference with the photosynthetic efficiency that I have read few of the study that was done by Simon Gilroy, and he has reported, because he is the one like father of astrobotany, I call him. So he has reported that 10% difference is there. But then, this will be a part of conversation. Like the question is why? Because it's more controlled. It's grown in a controlled environment, yet we see that difference. That's something that we can actually debate about. Yeah.
Okay, the other one is if terraforming can bring Earth environment in Mars, then how healthy environment that will be?
Terraforming of Mars?
Yes. Is it healthy?
Oh, that's. I think it will be more unstable rather than like leave on healthy because the way right now it is and the way we should understand life is whether we can form a Goldilocks zone or not, and the way the question has been put, the person has not taken that into consideration, so that will be a big issue.
Yeah, that's right. Your answer is right. It is not healthy. That is an answer.
Yeah.
Okay. So how does space radiation affect plant growth and phytomicrobiome?
Yes, and that's a good question. Recently, I'm doing something on a phytomicrobiome, and one of the things, space, not of the space radiation, but what I'm seeing is more affecting the phytomicrobiome is the environment that it's growing. Even though I am studying a phytomicrobiome, it looks like I'm studying International Space Station. Dr. Venkat, when you present your International Space Station study, it pretty much looks very much similar to that. So just to point out.
Okay, thank you very much for your time. These questions that is there in the Slido will be answered if needed. So we are tracking those in our database. So worries that if you're not having, you have the questions not answered, it's not your fault. Keep on asking questions. This is the time is we have to consider. Thank you very much.
Thank
Nitin.
you. Thank you, Dr. Venkat.
Yeah, similar to the talk that Nitin gave, we have the next presenter here, David Reed, who is going to talk about the engineering. Ten 10:30 to 11 is coffee break.
From 11 we started. I'll just inform.
What is this then? Okay. It's a coffee break here, so I could have asked the other questions there anyway. Coffee break should have been shorter. I'll consider that later. Nitin, are you still there?
Oh, I'm here.
Let us take one more question and we'll go for coffee break, okay?
Yeah, I can take two.
You can take two, but we cannot. Okay. Fertilizers for plant growth. If yes, how? The question
You broke up, Dr. Venkat. Can you repeat the question?
Are you able to read? Are you able to
Which
read?
one should I be reading?
The top one.
Okay. Do we use fertilizers for the optimal plant growth? If yes, how, what, and the chances that it will impact the microbiome of person consuming that plant. Okay, so right now, what we grow in International Space Station is not consumed. It was recently, last time that it was wiped with alcohol and it was consumed by Scott Kelly. Right now, we are not consuming. Fertilizers that we are using in International Space Station are NPK, very reasonable, the Hayes process fertilizers that are grown. But the only thing is, it's the larger crystal form that is being used in that because they want a continuous dissolving process going on there. And this is, I'm telling with regarding to advanced plant habitat, because that's what I have studied the most. Veggie, it's a substrate base. They have fertilizers already included in that bag of mass or substrate. It's not soil. Let me clarify that. There are no soil studies done on International Space Station. It's always substrate. And all soils are substrate, but all substrates are not soil. So just mark that point.
Okay, good. Thank you very much. Now we are going for a break, a coffee break where everybody knows.
Yeah, so the coffee break will be in the main dining hall opposite the auditorium towards your left. So it will be from 10:30 to 11. So we'll assemble here by 11.
Thank you, Nitin. And
enjoy your coffee. Thank you.
Any presenters not giving their presentation, please let them do it.
Yeah,
okay?
I think everything.
You got everything.
Yeah.
All right.
It's NPK. It's a larger crystal.
Still some people are there. We also want the audience in this auditorium to ask questions. There is no need for you to put it there, but if you're putting it there, that is also good. So that we can build a database. So whoever wants to ask question, 1 or 2 questions, I will be requesting you, and then appropriate speaker will answer that. So Shobhan is going to run with the mic, okay? If you are somewhere sitting in the middle, try to come outside so that it is easy for, giving the mic around. Otherwise it is too much, okay? Yeah, so when there is questions there, I will try to ask some information from you guys, okay? With that now, we still have 1 minute anyway. So thank you very much for the previous 2 presenters here. And one thing I want to say, we never use the word manned mission anymore. It is human mission. So we should give respect for both, men and women, because women are, growing more in space. So whenever you see manned mission, the speaker is wrong. It is a human mission, okay? I want to say that first. And also, the students, please try to mingle with the speakers and then talk questions during the break and other things. That will be, one of the opportunity for you guys. It is a tendency for us, the students who are friends, they form a group and then talk to each other rather than go and then meet the scientists who are visiting here. Okay, this is one of its kind of opportunity for you guys. With that, I'm trying to request Dave Reed here, and he's engineer. He's going to talk about the engineering perspectives on conducting space flight investigations. For example, you can think big and dream big, but that has to be appropriately realized, in other words, implemented correctly. So he's one of the pioneer of growing plants in space, and I knew him ever since he was in Kennedy Space Center. From there, he moved out to the different corporations. One of the right now he's the Florida Spaceport Operation Director for Redwire. Redwire is the company who won the space agriculture project. It's multi-million-dollar projects that they won. And NASA's effort to streamline spaceflight requirements and accelerate payload usage. In other words, you wanted to grow plants, you do all things lab scale, but when you are trying to do it in space, until it is different, it's going to be fail, or most of the time it fails. But this group of people led by Dave Reed has achieved a lot, and they if I'm wrong, they are the pioneer of growing plants and he got several awards. One of the thing is NASA's prestigious Silver Snoopy Award and Launch Honorary Award that he got. So here is Dave.
Thank you, Dr. Venkat. Thank you, organizers, for having me here. As Kat mentioned, I am an engineer, not a scientist, although I've spent the last 30 years of my life in the presence of some pretty bright scientists. So some of it has rubbed off on me. And we'll talk about.
Can you bring the mic closer to you?
Sure. We'll talk about the interaction of engineering and science today. All right. So mostly when we talk about growing anything in space, we're talking about model organisms. And this is just a little bit of perspective on the types of organisms that we fly in space. In the early days, and like in the space shuttle days, we basically took all comers. Anybody had some kind of an interesting idea. At one point, people talked about flying snakes in space, or specifically, we flew some koi fish on the SLJ mission back in the mid-90s. But things have kind of settled down now to the notion of using model organisms. And model organisms, of course, are things that are well characterized here on the ground. But more importantly for me, is they're things that are fairly well characterized, keeping them alive in space. Because as an engineer, it's my job to basically develop a life support system for whatever it is that's flying in space. And I've got some, just some typical model organisms that are used these days are listed up on the screen. Mammalian models are things like rats and mice. We often look at simpler organisms like roundworms or flatworms to model a specific component of higher animal physiology. And then, yes, because I'm in the presence of a lot of people that are interested in microbiology, we have a number of microbes that we study in a controlled manner as model organisms. But mostly today, I'm going to talk about plants. And plants are actually, in my experience, one of the most complicated things to try and keep alive because, unlike animals, say a rat, for example, if the rat doesn't like a particular part of the piece of equipment that we've designed for it, it moves to a different piece of the part of the equipment. If it's hot in one place, it moves someplace else. Plants don't have the ability to do this. As you know, a seed that falls, in a crack in the sidewalk on the ground is out of luck. I mean, it has to make do with what's there. It can't simply get up and move to a better environment. So plants have a lot of sometimes unanticipated coping mechanisms hard-coded into their organism. And as engineers, we're sometimes surprised in the way in which the plants respond. Dr. Sin did a great job of kind of setting the stage for plants in space at all. So I'm not going to dwell on this a whole lot, but from my perspective, engineering perspective, we're told that there are really four major things that we think about when we think of plants in space. First is the most obvious one, which is food, but not just food for us, and not the secondary thought of food, which is food for food, in the case of livestock, but in the context of the current discussions, food for microbes as well. As Dr. Sin said, sort of kind of standard decomposition that we're used to on the ground here isn't always set up well in a spaceflight environment, and that's something that we have to increasingly think about. Three other things that plants do. The next most obvious one that people think about is they revitalize the air. They draw carbon dioxide out of the air, and they provide oxygen to the air. Turns out plants actually have both carbon dioxide releasing and carbon dioxide consuming metabolic processes depending on whether it's day or night. And this makes it challenging from an engineering perspective because at certain times of the day, we have to consider the fact that the plant is adding carbon dioxide to its closed environment, and at other times it's removing it. And atmospheric balance, whether it's within a closed piece of machinery or within the larger space station environment, is something that's constantly being balanced by not only our own experimental systems, but those of the vehicle itself. Water cycling. Plants cycle water for free. Right now, all of the great big piece of Eclipse system that was shown in the previous talk, does one thing. It takes bad water and turns it into water that you can consume. Plants do this for free. I can give the roots of a plant a gray water stream. The plant takes that water up, transpires it off into the environment. All I need to do is condense it on a cool surface, and then I have water. So I get all that, not exactly for free from a resource perspective, but the consumables involved are considerably less than if you consider like a physical chemical system. And then something that's become increasingly apparent as we move to longer duration spaceflight are the physiological effects of plants. People sit inside of a metal can for months on end, never seeing anything else that reminds them of home, gets hard on a person. It affects an astronaut's performance. And so we find that people are very much interested in the simplest of things like tending a plant on the space station. When we talk about the researchers that want to operate in our equipment, we typically break them down into three large categories. There's the physiologists, and the physiologists care a great deal about what's happening kind of mechanistically within the plants. And then at kind of a higher order of organization are the crop people. The crop people, they would tell you, yes, I need to understand what's happening with an individual plant, but I'm really interested in just growing, so many square meters of plants, which of course right now in the current space platforms we have is not something that we can do. Have square meters available to us, but I can tell you that my company is in constant conversations with the next generation of the commercial low Earth destination space stations that are coming online. And there's a lot of discussion about having whole modules that are dedicated to plants, much more like an orbiting farm than like a multi-use facility that just happens to also grow plants. And then life support is one higher up level of complexity yet. The idea of a closed loop environment where plants are an integral part of resource cycling is something that is kind of a holy grail of people that engineer plant growth systems in space. The complexities are enormous. The resources have to be balanced, and we have this notion of equivalent system mass that comes into play. Sometimes when we talk about, how much mass of equipment is it going to take in order to conduct something? Is it worth the mass of equipment, or is it simpler to just take something that is consumed instead? Again, Dr. Sin talked a lot about what I call the direct influences of spaceflight on plants, microgravity effects, the orientation of the plants. It also affects a number of other things. There's these things called nastics and nutations, whereas the plants grow. If you've ever seen like a time lapse of a plant growing, it kind of corkscrews around a little bit. Turns out the roots do the same thing. They kind of corkscrew around. All of those things are influenced by gravity, and it's interesting in the way in which they, again, from a science perspective, how they get expressed in microgravity. Plants mostly perceive their environment as stressful. As Dr. Sin said, there are these heat shock proteins that kept showing up all the time. As it turns out, for the longest time, a lot of the perceived spaceflight effects of plants, or spaceflight effects on plants, were really, in fact, effects of the facilities in which we had put the plants, and not so much about being in space. There was an experiment done called the biomass production system back in the 90s, where the transparency of the equipment was much higher from a plant's perspective. And it turns out the plants grew almost the same as they did on the ground. This was a huge finding. And in fact, the researcher had to go before Congress and explain how is it that NASA spends all this money looking for spaceflight effects in plants when you're telling us that the spaceflight effects are actually not so much if the box is designed well. The challenges of designing the box are really what we're going to talk about quite a bit more here in the next few minutes. But these are these direct effects. Of course, radiation, the immune system of plants is compromised in the same way that it is in humans and others, because quite frankly, I mean, none of the living systems that are alive on Earth today developed in a microgravity environment. So it's mostly this perceived stress that causes this effect. But then there are these indirect effects on plants. So these are the things that as engineers keep us up at night. First is poor air exchange. You take for granted buoyancy-driven convection here on Earth. It's natural mixing of atmospheres. When I have a plant growing in a box, we tend to establish these boundary layer effects. First, boundary layers on individual leaves, boundary layers on canopies. You blow air at a canopy of leaves and it tends to just glide right over the canopy of leaves instead of perfusing the canopy of leaves. And at a local level, then I am told the plant perceives this as a stressful environment because the plant sees, say, a higher concentration of its own metabolic products in the immediate vicinity of its tissues. Root zone water management is probably the thing that causes us the most problems. The roots of a plant need to gas exchange with the atmosphere just like they do on the ground on Earth here. If you've ever seen, most plants don't like to have their root zones completely flooded, for example. When that happens, there can be very little gas exchange that occurs. Because capillary forces are kind of the dominant influence on water in the substrate systems that the plants are growing in microgravity, we have this constant challenge of over wet versus under wet. Many granular systems will nucleate dry zones that we simply cannot re-wet effectively space, not even with amendments in the substrates themselves. And then there are other things. The new car smell that you have anytime you get in a new car are the volatile organic compounds that come out of plastics, plasticizers, small, short organic chains. These things tend to accumulate in the spacecraft environment, and the spacecraft owners or managers only worry about those materials that are deleterious to humans, not so much to plants. So ethylene is a good example, which is a short chain carbon molecule. Humans have a very high tolerance to ethylene. Plants, ethylene is a very strong growth hormone. And so it influences things like ripening, like in bananas, for example, and the development of other parts of the plant. So we spend a great deal of time scrubbing ethylene out of the immediate environment in a lot of our systems. Water supply fouling. We get most of the water systems that we have involve some manner of a tank and some manner of plumbing that go from point A to point B. Biofilm formation is as much a problem in space as it is here. Getting rid of it or managing it seems to be more challenging in space than it is here. Sometimes that's just simply the case that we don't have the easy ability or the availability of crew time to remove and replace things that might be fouled. And then thermal performance is always a problem as well. You take for granted things like the computer that's in front of me right now just kind of naturally releases heat, and the heat, again, because of buoyancy-driven convection, moves away from the piece of equipment. That doesn't happen in space. Any areas of poor airflow tend to nucleate, say, a sphere or a region of higher temperature. That then affects the performance of the equipment, and then you can end up with a kind of a negative feedback loop that shortens the life cycle of the equipment. So when we talk about engineering for space flight, this is kind of my list of things that I need to worry about. The first, excuse me, and most important is crew and vehicle safety is number one. If the vehicle isn't going to be safe, if the crew isn't going to be safe, the simple truth in our cold hard engineering facts is whether or not your science is successful is third place. And so we must absolutely make sure that things are safe. And conduct of a safety assessment in the context of human spaceflight is very different than what you would do, say, if you built cars on the ground or something like that. Second is that resources are super scarce. The amount of power that this laptop runs is sometimes 3 times as much power as I get to run plant. We have plant growth facilities that use upwards of 1,000 watts. We've had some that use 20 watts. Talked about materials a little bit, the suitability. We have to worry about not only are these things flammable, what are the combustion products in the event that they do catch fire. Smoke is bad, caustic smoke is worse. for a crew member. So we have to worry about that. We worry about the growth substrates. We already talked about the fact that actually Dr. Sin said that people don't fly a bucket of dirt in space for a variety of reasons that we don't have time to talk about. But these substrates themselves have complexities that make them a challenge to work with. As much as we can, we try and remove the human from the human spaceflight environment. What does it mean to do that? What it means is that people make mistakes. When you have an astronaut that's on orbit for a 6-month stay and every single minute of his waking or her waking day is prescribed to him, it's not that it gets monotonous, but it gets just mind-numbing to try and keep on task. And I've had astronauts make very simple mistakes that very nearly either broke my equipment or cost mission success. So as much as I can, simple things like having 2 different sizes and types of quick disconnects to put water into one reservoir versus another reservoir will keep an astronaut from making a mistake and putting it in the wrong place. We've had, and these are bright people, that we fly in space, but they make simple mistakes because we're all human. Data storage and retrieval is a challenge, especially with living systems and things like plants, because today we don't have a lot of analytical tools in space, nor do the astronauts have the skill set in order to do those analyses in space. And so right now, at least in plants and mammals, mostly that means things have to come back. And then operational constraints. You can't turn over seeds or plants weeks or months or a year ahead of time the way you can with other types of pieces of equipment. The operational environment is dynamic and changes every single day. We think we know going into an experiment exactly how our equipment is going to operate and how the plants are going to perform, but then we are surprised every single day. And so for something like the chili pepper growing experiment that we did on the space station, it went for 120 plus days. That's 120 plus days. Holidays aren't off, weekends aren't off. The plants don't care, the hardware doesn't care. This is kind of a survey of pieces of equipment that I've had anything to do with in the last 20 some years. You'll notice that most of them have kind of a form factor of a microwave oven sized box. And that's a heritage from, a heritage from the space shuttle days. And these microwave oven sized boxes is something that is the same form factor is moved forward to the space station as well. Some of these things are very simple cylinders, like you see in the bottom left, where you can't even see what's going on inside and there's not much going on, all the way up to powered facilities that you can see have grown everything from grasses to trees. These are the 2 current facilities that NASA has on ISS. They do very different sorts of things, even though they grow plants. The first is Veggie. It's a low resource box. It's essentially just lights and a small fan. But with lights and a small fan, you can do a considerable amount of research. But there's all sorts of variables going on. The temperature changes all the time. Crew member exercises next to you, and so the carbon dioxide level goes up. So doing single variable science in Veggie is very challenging. And the second is advanced plant habitat, which is a refrigerator-sized powered box that does it all. It manages the atmosphere, it manages water, it recycles water to the plants. My company is responsible for managing plant habitat for NASA at the present time. Briefly then, here's some pieces of equipment that my own company has developed. The first is called Ponds. Ponds is essentially, in a nutshell, an open box of water that has a flower pot growing inside of it. And through a variety of engineering that I would love to talk to people about in the offline, we basically convince the water to remain inside of the open box of water so that it doesn't just come out into the spacecraft. But that provides an almost constant flow of water to plants growing in this system. And you can see there's a picture of a Mizuna plant growing in the ponds on space station. Second is phytofuge. So we go to space not only to test out microgravity concepts, but before we get to the surface of the moon and the surface of Mars, it'd be nice to know how exactly model organisms are going to respond to that environment. So we have a small centrifuge that is outfitted with a variety of these kind of trapezoidal boxes. This particular one grows plants. so that we can spin at lunar or Martian gravity, or have an on-orbit unit gravity control at the same time that enables us to interrogate the plants on the space station. And last, this is plant habitat. As I mentioned, since 2018, my company has been responsible for managing. We've grown everything from, you can see, radishes, chili peppers, and more recently, we've grown some callus cultures as well. And just a minor correction from before. So both these chili peppers and these radishes were consumed on the space station in the last couple of years. Lastly, looking to the future, all these systems still have relatively small footprint and relatively small growth height. We're in the process of developing what is essentially an erectable tent, a grow tent that will go on the space station in the Cygnus module. When the Cygnus is a visiting craft, it allows us to open this tent up, grow very large plants inside here, and then deconstruct it and push it to the trash. And then, this is just pointing to the future. We hope one day to find ourselves on a space station that has lots of plants growing in it. And boy, I would love to have this kind of, this fiction in the lower right-hand corner is actually, I think this picture is from the 1970s. But one day we'll get there. For the record, my presentation has a number of slides in the back, which we don't have time for, that just shows some other pieces of equipment. Since this is going to be posted, you can, of course, ask questions about all of these, either in the chat or ask me later. I'll be happy to answer them at another time. Thank you.
Thank you very much. David, it's a wonderful presentation. A lot of people who have seen the movie Mars may come and ask you about, how Matt can grow, potato out of his own, stuff. And get ready for that. And let us, anybody in the audience wants to ask questions, please give the mic to her, please.
So the model organisms, I think of it as kind of like crowdsourcing. It is the investigator community themselves that define the model organisms. So, for example, Arabidopsis thaliana, this small plant, is very well established in terrestrial biology, and so as a collective, the plant research community decided to use that as a defined model organism. I just work with what they give me. It's kind of the way I would put it.
Thank you. So the online question here is there. The audience also here, Anurup. I think this question may not be directly related to him, but still, let's ask: how do you scrub out a specific gas from a closed environment like ethylene?
So we have these scrubber modules. So if you can imagine in a closed environment like the plant habitat that has this completely closed growth chamber, we can pump air through a series of small. I'm a chemical engineer by training, so I'll call them a packed bed reactor. That's what they are. And in the packed bed reactor, we have granular material. For ethylene, we use potassium permanganate impregnated spheres. And the potassium permanganate then strips the ethylene out of the atmosphere.
Any question here? Go ahead.
Hi, it's a wonderful talk. So my name is Tulika Srivastava. I just want to know that have you observed any metabolic changes like gene expression changes in the plants that you're growing in this kind of facility, or those which are grown on a normal environment, under a normal environment?
So the answer is yes. Again, engineer. So I'm going to go out on a little bit of a limb to talk about what the physiologist would tell you. But yes, gene expression changes are common. I think Dr. Singh made mention of the TOAST database is a good place to look for Arabidopsis, especially. So yes, it's quite common.
Yeah, there are a lot of work done on that, and if you log into the Gene Lab which
Yes.
is a database, have all the experiments listed, so you can learn from there and design a new one that has not been done. It is open to international community. It is not, restricted only to US citizens. So you can get all the data that's cleared by NASA and placed it there for future scientists to design new experiments.
Thank you.
How is the next? Has NASA considered using algae instead of plants to generate useful biomass in space?
Yes, and in fact, there are a number of substrate systems being considered to grow algae. I mean, there have been some simple algae experiments, but mostly using algae as part of a miniature closed environmental system. There was something called the CEBAS, CEBAS, that was flown by the German Space Agency that involved some algae, some snails, and some fish, all operating in a marine environment, in a water environment. And there have been some other attempts at algae as a substrate, just on a substrate growing under photosynthetic light as well.
That's right. The Indian Space Organization, ISRO, also flew the Japanese experiment with the algae Synechococcus to see what are the effects of. Okay, there's another question.
Yes, sir. This is Jayashree. How does the food deterioration process differs from mass to mass, sir? Food deterioration process or preservation?
So food deterioration process. I'm not sure if you're talking about food that we take.
Preservation, you grow them and then you preserve them for a long journey.
So if we grow them here on the Earth, if we grow them in space. So, well, likely it would be the same way that you would preserve food on the ground, which is to say either to refrigerate it or freeze it to slow down degradation. I mean, it turns out that, the same sort of microbial communities exist, and Venkat's going to talk about this, on the space station or in spacecraft as occur here as well. We still get rotting of food exposed to air because the air stream itself in the space station is not clean. It's no more clean than here.
I'm going to go one more extra because the name is Shreya, a student, women student should be answered here. If a plant is grown in a closed module, how do you ensure optimal conditions for temperature? It's the last question.
So I mentioned that in a closed module, we have to force convection. We have to move air with fans in order to get it to where we need it to. Much in the same way that your air conditioning system works here. I mean, the air conditioner for this room is not in this room. It's somewhere else. And there's ductwork that moves air from here to some sort of a heat exchanger within the air conditioning system, and then it comes back here. We have the very same sort of thing, but on a very small scale, where we have a system that can either add or subtract heat to the airstream. We pull air from the module itself, flow it over a cold plate or a reversible cold plate, so it could be a warm plate, and then back to the chamber.
Thank you very much, Dave. And a round of big applause for him. And our next speaker is Dr. Fatih Karuya. I knew him quite a long time. I know when he was a grad student with Dr. George Fox lab, later he joined NASA and he's still working in there as a senior research scientist affiliated to the Blue Marble Space Institute of Science within the Astrobiology and Space Sciences Division at NASA Ames Center. So he conducts research related to astrobiology, space biology, and human space flight. In particular, he's interested in life beyond Earth and how biological systems respond to the space environment. He's interested in understanding and mitigating the effects of long duration space flight on astronauts' health, developing high throughput technology for in situ space research. Dr. Fatih has successfully implemented 18 investigations using model organisms or human aboard the space station to contribute the research associated with space exploration. Here is Fatih.
Thanks for the introduction. It's a delight to be here. I just want to thank the team for having us over, and in particular, Karthik Raman and Katsuri Venkateswaran. Actually, everybody calls him Venkat because nobody can say his name properly, but I am one of the few in the US that can do so. So again, thank you very much. That being said, I wanted to kind of also disclose a few things. I do work for NASA as a contractor, but I'm not here on behalf of NASA, so that's one important element. Secondly, I'd have some disclosure, one of which is an entity for which I am a funder, which is called Space Research Within Which. And then the last thing is that during my presentation, I would be talking about specific lesson learned and how things fails in particular. And that was part of some project I was involved in biology and with some commercial partners. And so here it's kind of trying to emphasize the learning curve. It's not here I'm not here to blame anyone. And as I say, I was part of so here the emphasis is on the lesson learned as opposed to blaming anyone. So here's the outline. I would be kind of give you an overview of what has happened on board the International Space Station for the past 22 years. As you were well aware, I mean, we have a human being on the station orbiting around Earth for the past 22 years. And so obviously a lot of things had happened from a scientific perspective. That being said, I will be kind of giving us a brief overview of what are the current trends in regards to the type of payloads and instrumentation. We had a nice presentation by Dave talking a little bit about some of the instrumentations in regards to plant biology. I will be expanding that a little bit. And then also moving kind of forward, what type of payload and instrumentation are needed for us to be able to have a meaningful type of investigation when we are starting to kind of go beyond low Earth orbit. And then, as I mentioned, some considerations. So here are the kind of publication highlights coming from the International Space Station. So it's kind of a year old, so it was up to 2021. So roughly there have been over 2,500 publication on peer journals that comes out from investigation conducted. So up to date, there's roughly 3,300 experiments that was conducted on board the International Space Station. And here it's breaking down by different categories, some of which are related to human health, others to space biology and biotechnologies, and then technologies, Earth observations, and so forth. And so the key aspect here is that you have a large portion that are related to human health and also for space biology and biotechnology. The interesting part is that you can see from a trend in regards to publication to the top 100 journals is that that trend is kind of increasing nicely, kind of exponential curve, which is very nice to see. But similarly, you have a very large amount of publication which are coming from the biology and the biotech world. So as you can see, this is an exciting time for all of you to be involved with those type of research. When you do a network analysis of specific keywords coming from all those specific publication, if you look at what happened from the past decades, going from 2000 to 2010, you can see some keywords like microgravity, space flight, radiations, and so forth. However, as you can see, the center of inertia looking at that particular network from 2011 to 2020, mainly focused now much more on human health, much more on space biology. And as you can see here, keywords are more expression, gene expression, the immune system, and so forth. So as you can see also, the whole field is moving more from fundamental biology to more omics-like or omics type of biology, which was obviously very, very exciting. In regards to current type of payload and instruments which are available, and so the table is kind of divided into two categories. The first one is related to the ISS, and then CubeSat. So on the ISS, obviously you're studying the microgravity environment. You kind of orbiting around Earth, so you're not too far. The radiation level is, yes, higher, but not that high. And then obviously you have human attendants, and so crews are present, and so they can help for the implementation of any particular investigation. And which is kind of very different from CubeSat type of technology. This is a nanosat, and so this needs to be automated, much smaller in size. However, then you can reach higher altitude and therefore higher exposure to radiation. Then you have similarly free flyers, and so those could be for model organisms going from microbes to animals. And then moving forward from leaving low Earth orbit to going beyond low Earth orbit and the Artemis mission in particular, now you will have those different platforms. The gateway, we can have platform and payloads inside the habitat. It could be also outside for a particular subset of experiment where you look at exposing those samples to the harshness of the space environment. And then lunar activity, lunar surfaces. And so you will have those different payloads. Certainly, these are still TBDs because we're currently working on those requirements. But as you can see, there's a very, I guess, nice aspect in regards to the complexity. And the key thing there is that when we're moving to the Artemis mission, the crew attendance will be much lower than it is currently for the International Space Station, which means that automation and miniaturization will be key. In regards to the type of investigation, when we involve, and again, this is kind of NASA-centric, but nevertheless, there's also international partners in this graph, is that 3 different type of categories. One is a NASA-owned type of devices or payload. The other one is commercial-based, and the last one is through international partners. And so you have like devices that you can use as a plate. For example, you can grow some cells and bacteria in particular. I will be talking in a minute about the microbial tracking. And so here you're just swabbing different surfaces on board the International Space Station. From a commercial perspective, for example, here I'm talking about the fluid apparatus system, just simply a tube where you have different chambers within which you can have your samples and you can have cultures. And then if we move to different type of instrumentation going up, for example, obviously you need to have some incubators because you need to have some specific type of temperature. And so for that, you have a suites coming from the commercial partners. And here I'm just like, for example, giving an example coming from one of our close partner, which is BioServe. And so you have devices which enable you to have a gas exchange as well as specific temperature, which are needed for specific sets of experiments. Then if you go for more complex type of model organisms, like for example, for cell biology, and so NASA do have some system, one of which is a bioculture system, but then as Dave just mentioned, also Redwires has a few sets of instrumentation that enable cell culture as well as a plant biology type. And so as you can see here, there's a full spectrum of devices, not just from the NASA side, but also from a commercial partner. And as I say, obviously the ISS is a station, but it's an international space station. And therefore you have also other partners coming from the European Space Agencies as well as JAXA, which in particular, which also do have different type of hardware and technology that could be used as to perform those type of investigation. What is critical these days, obviously, is to try to have specific control, in particular devices with a centrifuge, so that it enables you to kind of compare in-flight microgravity to 1G or partial gravity. And so there's a few subsets of devices which are amenable for those type of research. So here I'm giving you a video, and so it's coming from the crew that's coming from one of the experiment that Venkat is involved with, which is Microbial Tracking 3. And so I'm just, here it's pretty straightforward, just like someone just swabbing an area. But as you will see very quickly, one, it takes a little bit of time, and two, it's more challenging. So one, the crew, which is workers, is cleaning herself because she will be sampling from the wall here that you see in here in the background is the cupola. Two, she will be taking a bag within which you have the wipe, and so she would be cutting the bag to kind of take out the wipe, and then see what happened to the scissors. The things are flying, right? So I have to pay, you have to pay attention to those type of things. But as you can see, she's kind of putting all those plastics all around her belt, and there's some Velcro there so that those things are not flying away. And so here again is the idea there is to kind of appreciate the fact that, yeah, I mean, just swabbing a surface kind of looks simple, but when you do that in space, you can see it's more challenging. So here she's going to grab the gloves, and so she will be putting her glove, and actually she's doing a pretty good job. So you will see that in a second. And so again, to try to minimize contamination from the crew, we require the crew actually to wear sterile gloves. And so like that, when the crew is sampling the surface, we know the microbial community is mainly coming from the surface as opposed to come, from the hands of the astronauts. And so now she would be taking the wipe, and now she would be starting wiping the surfaces just like outside the cupola. So we, ask her to do kind of 3 different direction, going side to side, then we'd be up and down, and then going from one corner to the other. And so anyway, here's just to kind of emphasize, even simple tasks kind of require a lot of things. As you can see, there's all those bags flying around and so forth, and then you need to maintain sterility. So as you can see, this is, and again, this is a simple mission. So let me give you another example, which is micro 11 I was involved with. And so that required 2 things. One is that you have some media, you have some cells, you need to warm up the media, you need to warm up the cells. And then there was some activity inside the glove box. And so as you can see here, there's 2 things that needs to be done. Actually, that experiment required 2 crew members. And so it's already extremely complicated to get access to one. For this one, it requires 2 crew members, which is, requires a lot. And so the crew members here, so one of which was Serena. She was doing the work inside the glove box, and then Pistol was doing all the microscopy. And so here is one area of the station, and you will see the good thing about the station is that you have some racks and you can move things around. And so all the things were kind of set in a way to kind of optimize and to limit waste of time from the crew going from one place to the other. And so again, here you have the glove box, you have a microscope, which is here, the camera, which is here, you have the software, which enables you to perform the video. So you have the cassette there where you would be observing your samples. And then you have a freezer just up there. So you can do your experiment, and then you can put your samples up there. And then you have like an incubator, which was used to actually incubate some of the samples. And so you see here, you have like an environment which is enabling you to kind of in an optimal way minimize the wait of time to kind of go from one place to the other. So I have a video, but it might take a little bit too much of time. It's kind of briefly showing her, working inside the glove box. As you can see, it's not very easy. It's kind of bulky, and actually crews kind of complain always when they're working inside the glove box. But anyways, here's kind of more complex type of configuration. So going from one hand with a microbial tracking 3 to this type of investigation requires much more work and much more type of capabilities. So ThinkRare is like test what you want to fly and then fly what you tested. And so here we're going to talk about 2 things, one of which is one experiment which I was involved with, which is MVP cell 2. The idea there was to use Bacillus subtilis and do long-term studies. And the idea there was to have to use some spores, and you grow them on a track, and then you collect them at the end. And so here are the type of cassettes. And so here you have those cassettes, you have the track, and then the media was here. And the idea is that you activate those samples by pushing the media into the cassette, and that starts the activation. Here is the configuration where you have all the samples inside those specific cassettes. And actually, this was done during the EVT. So EVT stands for Experimental Validation Test. The idea there is you test your samples before you go to space. And so when we activated, for example, this specific cassette, you can see that actually there was a backflow of media back into the syringe. And so that was an issue for us. And so what we decided at that time is that actually we had only 1 check valve. And so the decision was made to kind of add 2 check valves to kind of minimize that backflow. And so here I'm going to show a video of the crew, which is here, donc, using the cassettes and taking 1 cassette and trying to activate one of the cassettes. So the idea there again is to kind of inject the media into the cassette so that activation can occur and the spores can grow, and then they can grow among the track. So as you can see, he's trying, and then you start to see there's some resistance there. And he has a piece of paper because actually there's some leaks starting to come out from the cassettes. And so then, it's kind of questioning, okay, how am I going to do that? And so, we were talking to him, trying to kind of troubleshoot the issue with him. And so then, he's going to take the cassettes out. And then you can see he's going to try to push very, 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 very hard to try to kind of transfer the fluid into the cassette. You see the crew here. So it's kind of pushing, and you can see he's pushing hard, and still nothing's happening. And so that led to a few things, one of which was the fact that, as you can see, cells were growing, but they didn't grow up until the dot there, which is what was the angle. And then we were doing some fitness studies, and so we were taking images at a particular time, and not having, full amount of media going into the cassette was an issue. And so actually, currently, we're working on reflying that particular mission. And so again, here, I'm not trying to blame anyone and anything. As I say, I was part of the problem as well. But that tells you that, you know, when we decide to change from one check valve to two, I mean, we thought, well, yeah, you know, it's not rocket science, it should work. I mean, that was tested and all that. But the thing is that those cassettes stayed in space for quite some time. And then actually those having two check vials led to the fact that actually we were not able to optimally activate those cells. Another example here is Micro-8. And so Micro-8 was an investigation, also space biology using Candidas albicans. And here we're using group activation pack. So which is a device which is kind of pretty nice because it's pretty quickly you can actually activate all samples here. There's 8 samples, so the 8 barrels that you can activate at once. And so here you see all those barrels there, and the idea there, you have your cells there, and then you can transfer fluids from one chambers to the other. And so here we have 2 crew members, one of which is holding the gap, the other one is like cranking it, and so that enables the activation. And so the way it works is like, for example, you have your cells, and then you have a chamber with media or chamber with, for example, a preservative, and then by cranking it, then you're transferring the media to where the cells are, and therefore those will be able to grow. And then later on, you can fix them if you want to. What happened in this particular experiment is that one of the industrial partner, which was providing the septa, which is inside that fluid apparatus Borel member, actually changed type of providers. And so we did some tests, different type. Actually, the company did a lot of tests using different type of microorganisms, and looks like to work fine. However, when we performed the activity in space, actually no cells were growing, and the troubleshoot came back, and so we did the assessment, and then we realized there was an issue with the septa that we were using, and actually that septa was bleaching out inside of the growth chamber, and that was inhibiting cells from growing. And so that was obviously also another type of issue. And again, here we thought, well, yeah, we can just change the septa, everything going to go well, and actually that was not the case. So when you're in space, there is some issues. And so some issues are the fact that, you know, on Earth, you have multiple type of replicates, multiple type of samples. I mean, you're the expert on all that. Of course, in space, that doesn't work where you have limited resources, limited capabilities. And so certainly this is an issue. And so when we move forward from low Earth orbit to beyond low Earth orbit, yes, you know, we've been in space for quite some time, some of which are stay for over a year. We've been to the moon only for like 10 days or so, but then when we're going to go back to the moon and stay for 30 days, then we're going to go on the lunar surface and onto Mars, all those things are still unknown, which means we need to do more. And so all those type of risk that we are currently working on, and certainly radiation is one of the main limiting factors. But then we need help from technologies. And so again, you know, 19th century was engineering, 20th century was chemistries and physics. Now we are at the edge of biology. And so here's the central dogma of biology. By having specific type of instrumentation, you can inquire about DNA, inquire about gene expression, and so forth. Then when you combine all those type of analysis, then you can discriminate a healthy individual from someone which is not. And so certainly those type of methods are very, very appealing. The problem is that there is some issues with space, and in particular, the fact that it takes a very long time to send an instrument in space. More importantly, I always give you an example here. Here, for example, it is a type of hardware which is RT-PCR, so nothing fancy, came out in the market in 2000. And we took the same device on the station like 16 years later. And so as you can see, there's a big gap between things coming out in the market and things being used in space. And so we need to do better. And probably mini ions is one thing that enables us to move forward. And so here I'm showing Kate Rubins. You know, she was the first one to sequence DNA in space, but still, as you can see, there's still like processes that need to be involved to be able to perform those activities. And so Venkat and his group have been working on technology. He'll be talking about one of which later on, so I'm going to skip that, but I will be talking about one of which I was involved, which is GEM, which stands for the gene expression measurement module, I'm sorry. And so this is like a 10 by 10 by 10 device and perform one-to-one to end analysis. So one, you're able to grow cells, extract cells, and then hybridize it on a microarray. So we did some testing, we did some validation, and actually we show that one is extracting nucleic acids, and as you can see, the quality and quantity is similar to control, but more importantly, the expression profile was very much similar to what we do see when we compare that to microarray platforms. So moving forward, on one hand, you have commercial-based platform, on the other hand, you have space-based platform. Certainly, the advantage was, you can have automation, you can adapt it to space, but that might require some space qualification. On the other hand, when you have from the get-go a space-based platform, that will require long time to kind of develop and test and validate the platform. Maybe you have something between the two. It could be a hybrid platform, and so kind of plug and play. And so again, there's a lot of consideration. However, I do believe that moving forward in the next few years, you can have those type of technologies, and in particular, sequencing with mini ions, maybe some microarray based and PCR based. Then in the next 3 to 4 years, I mean, certainly, having proteomics and metabolomics are things still that require some time. But I think those will be, needed type of instrumentation for us to kind of have more meaningful type of analysis being done in space. And so conclusion here is, as I said, I mean, within the next few years, we can have all those omics type of capabilities, and those are needed so that we can increase the type of discoveries as well as the type of breakthroughs that are needed, not just for us on Earth, but also for exploration. So I will stop here. I will happy to kind of ask you to ask any questions, and then certainly you can, follow me on those social medias. I post a lot of things related to students, postdocs, and information. So thank you.
Thank you so much for a very, very good talk. Just to give a thought, as I said before, we have to dream big, but that has to be appropriately implemented. So now he has shown a snapshot of the issues and the problem of getting a sample, which is in our mind very, very simple. But there are a lot of things there that, it is not as simple as we think. But the back end of developing detection methods and instruments are having a lot of research to develop. But the front end, which is a simple sample collection, is not there yet. Okay, so the young mind has to think about it next, what we can work on it, and then we can simplify all this process. Okay, with that, is there anybody here want to ask a question? Please give the mic to her.
Thank you for your talk. With you showed the plans for at least 15 years. But does it mean that NASA is going to continue with ISS because Russia is pulling out now, and even was threatening that they will crash the space station? I hope that NASA is taking over the control.
I mean, certainly, I'm not here to comment the news, but I think currently all international partners are going to work with each other, and currently the set time for the International Space Station is 2030. So we still have a few years. And then, as you probably were aware, commercially platforms are coming up online. So there have been few sources of funding which have been provided by NASA to commercial partners. And so there are a few platforms which are currently in design. And so there's Axiom, there's Sierra Space, there's Nanorex, and so forth, Starlabs, and so forth. So the idea moving forward is to kind of have some time where you have commercial platform as well as NASA orbiting around Earth at the same time, so you can have overlap between the two, and then you have a takeover from commercial platforms.
Thank you.
Okay. The other question, let's take it from here. How many of the existing hardware and solutions are adaptable to the new planned space station?
So I'm assuming your plan is talking about commercial platform. And so currently, again, there's a lot of conversation between all the different partners and NASA in particular to try to kind of provide some specific requirements. But you don't want to kind of provide too many requirements. I think the idea there is that there's a lot of things which are unknown. Certainly, there will be a lot of things which have been learned from the ISS heritage. And so that will be taking in. So for example, having those racks, which is kind of very nicely enabling you to kind of move things around, and certainly that's amenable, but certainly the door is open. The door is open to new type of instrumentation, and I'm hopeful there will be those type of analytical type of instrumentation, which enables you to perform analysis in space and not just being in a way where we're currently conducting science, which is sending something in space and then have to wait until samples return to the ground. And so I think there's room for innovations, but certainly I do believe there will be an overlap between the technology which have been developed specifically for the ISS and those specific commercial platforms.
Anybody here? One more? No questions, so let's go here. So, is there any research on bioprinting of human organs using mammalian cell cultures on the space station?
Yeah, so there's 3D printings currently happening on the station, and so certainly printing tissues is very much appealing, and the reason that behind it is that you have a lack of gravity, and therefore you enabling a better structure to be built from your printer. And so certainly there's a lot of interest in those type of area. And so, yes, definitely this is ongoing, and similarly, you know, organ on the chips. I mean, lately this is something which a lot of people are working on, and there have been, you know, a few subsets of experiments and investigation which have been performed in space in that regard.
Any question from the cut?
Hello, sir. So I just wanted to ask the question. So the samples collected in space get like contaminated in slave. So what kind of contamination does happen?
I'm not sure. Which type of samples we're talking
No,
about?
any kind of sample that you collect, are you getting that get contaminated?
Oh, that happens often. You know, first microbial tracking 1 mission, we had an agar plate. That was, so we call it, so one for bacteria, one for fungi. And so we had specific procedures describing how to use the plate. And more or less, you just remove the sleeve, you place it on the surface, and then you just put the sleeve back, put that into a bag, and then send it back to us. So when we look at the agar plate, actually there was fingers on it. And so what happens, the crew didn't know which was the right one. So you remove the sleeve, then touch, oh yeah, that looks like an agar plate, and then put the agar plate on the surface. So yes, there's contamination. But you know, you saw from the video, I mean, we tried to, you know, ask the crew to kind of wear gloves and things like that. But you do have also control. So we send wipes. If you're talking about wipes or swabs, so we send them into space, and so we do also have control where we wipe the air, and so as a positive control, and so we do try to kind of minimize those, but at times things happen. And so
we are over. So these 2 questions, like, you know, gut microbiome, we have a session tomorrow. Stefan Green is going to head it, so you can hear more about that later. And MinION sequencing, data processing, we will talk about that, you know, answer that in an email. Thank you very much. Give her a big round of applause for Pat.
thanks.
To you now.
I think Venkat's already been introduced. But still, let me just say a few words. I think Venkat's gone a long way to get this exciting session set up and bringing a lot of collaborators from all over India to this meeting. So I think, you know, we all owe thanks to Venkat. And I think you've all seen his CV, and he's got closing in on 50 years of research on various aspects of the microbiome. And I think the students should really, as he was suggesting, try to grab some coffee with him and talk to him for questions and insights. So with that, let me invite Venkat for the keynote. He's going to talk about microbes in space, issues and solutions, about omics in space.
Thank you, Karthik. And I think I need the color mic, because I won't stand in one place. on. You can switch this off, right? Otherwise, it will give some echo. Are you guys hearing? Hearing? Cool. So is it clear? Hey, can we dim the light if we can? The white light. It's easy for people to sleep also. You cannot dim it? Are you guys clearly seeing it? Okay. Okay, so this, what? Okay, this is our space. It's coming, going and off, right? Hello? Okay. This is our jet propulsion lab. So first I put this one because it's not coming. Come and goes. Why? Right? It just comes and goes, right? Cell phone. Okay. This particular slide shows 3 different generations of the mission. Okay. This is a Pathfinder mission, which is smaller, 1 kilogram level of the rover that is that rode Mars. Okay. 2, Phoenix 2 MSL mission. So this is MSL mission is nothing but the same Mars 2020 also, that is similar rover with a different science instruments, but the weight is roughly 1 ton, which is a Mini Cooper size. So this is golf cart size. So as Dr. Bernardini pointed out in the beginning, for the planetary protection point of view, the allowable microbial burden is same, whether it is 1 kilogram level or 110, tomorrow 10, I don't know. That means he already showed up in one of the slides, allowable bioburden is 5 times 10 to the 5 spores. If you take 1 liter of orange juice, you will find more than that kind of microbial burden. So you also saw several geometry of these things, so that also is accounted. So the cleanliness level is same. Today, I'm going to talk about challenges and solutions. Finding the problem is not going to serve the purpose. And we have some solutions. So I'm going to touch a few things from extreme environments, deep sea to the space station, as well as what is associated with the rovers, like a hitchhiker, like a passenger list that is going to the exploratory planets. Some thoughts today. Why microbiology in NASA? Right? Why do we need to do microbiology in NASA? Because it's the engineering institution. Because we are dreaming big. The question is, are we alone? If you're asking such kind of questions, are we alone? The biology comes into play. Like, we get implanted from other planets, or we evolve from physical and chemical reactions, by that combustion become biology. So that is called astrobiology mission. That's why, biology kind of questions are asked. If you are not asking such a big question, then things will not move forward. To get your dream realized or implemented, 99% of the engineering concept needs to be done. Okay, so planetary protection has already been introduced to you. There is a robotic mission. But tomorrow, if you have human in space, you need to take the friendly microbes with you, the beneficial microbes. And we are made up of microbes, basically. 100 times more cells, microbial cells compared with our own cells. What that means? That means we cannot live without microbes. That means if we walk into other planets, being a walking fermenters, we shed them. You have to make sure that all the microbes are not bad. Without microbes, we cannot live. So we have to live with the microbes, whether they are doing good for you. If it is making good stuff, yes. So you have seen people talking about the plant science, talking about the other animal-related science. All require microbes, beneficial microbes. So you can study that. That's what space biology means. That relates to the human research program. So while doing this, you come to know a lot of microbes that you have not come across yet. That doesn't mean it came from Mars or some other places and then get into the International Space Station or something that we are finding that as a novel bag. We might have taken along with us either through the human or through the cargo, the experimental cargo that you need, the experimental animal you need, right? Those are not transgenic material. They have inherent microbial composition. Then why we get more novel microbes there? Because relatively space station young, only 22 years old. So you will be able to find such things that you're taking it, but you go back to your garden soil, you know only 1 to 10% of what is in there. 99% of them that we do not know in our own backyard. But the thing is, are they doing something useful so that you can describe them as a novel? So by characterizing those novel and unusual microorganisms using various different technologies, you come to know the problem. Is it doing any kind of harm, like a biofilm formation? Is that increased under the microgravity? Can we use genome, proteomes, and metabolome to understand that? Sometimes you cannot isolate all the microbes because it requires different kind of cultural conditions, as we know. But metagenome, all of you what they are, from the metagenome you can tweak and then regrow the same samples and then find the one that you need. Or in the archived materials, you can go back and then look for the one that you missed. This is the animation for you to have some thing, what we are doing right now, Mars 2020 rover that is drilling and how it is caching the sample right now. So that gives you some information about what are the issues that you may face if you are looking for life in other planets, how important collecting samples are. This doesn't have any audio that much, so that's why I'm going to talk about here. Think about these rover wheels. You have to clean them. Why the people are calling me when I'm talking? Okay, so see this kind of stuff. So different, geometries. All these things needs to be cleaned. Otherwise, this leaves a footprint of what it has taken along with that. Now, this one is going to go and then look for things and then drill, and then this drill has to be cleaned to the level that you cannot detect, below detection limit. That collects the samples and then analyze with some sort of analyzer, looking for the geology of it with some sort of, spectrophotometer and other things which is made. All these instruments and that it get delivered has to be appropriately cleaned. This tube, the drill holding tube, sample collection tube, has to be cleaned. We do not know now what is Martian life. Can it withstand 150 degrees centigrade? That we don't, that our life not exist anymore. But we take NASA, go beyond that and bake them to 500 degrees centigrade. If you are baking 500 degrees centigrade, the whole instrument, whole rover will be not functional, right? So materials that are not compatible to that height, but the subsystem that is going and touching the precious sample, which is what, if I'm wrong, you can correct. Roughly 2.5 to $3 billion worth of mission will be gone. So that's why the subsystem that collect the sample and catch it are cleaned to the level 500 degrees, sterilized to the level 500 degrees centigrade. So today I'm going to touch about some of the space facility as an example, and how you can do sequencing in space automatically from sample to sequencing, not only sequencing. Okay. I'm going to talk because Stefan is having its own, session. And application of some of this microbiome research, like I will give you a story of the Kalamiella and Methylobacterium ajmaliae for plant growth, and computational biology, Klebsiella story that I was working with, Karthik's group. First, space microbiome. I'm not going to touch everything because of the lack of the, thing, because all leads to the space station are from space station. Some of the things that we are using it for, through resupply vehicles, and we have a mock-up of the space station minus microgravity as a ground control, and we have inside, and all the data can be mined, and some of them you can intentionally take and then see what are the drugs can be appropriately discovered, and then this real estate is so huge, outside is there for 22 years. Is there any microbe that survive outside the space station? And we can intentionally take and expose to the outside the space station. I am not going to talk about today, but tomorrow or late evening today, Onofra is going to give that kind of thing. But now let's talk about the space station inside, and later on omics in space. So this is us. Anybody knows he is the one who spent 350 days and almost a year in space. He's the one who spent longest part of the longer duration of humankind. And Terry Wirth is the one who sampled me. See this is brownie they're eating. That means you have penicillium along with it. Okay, so I'm not going to detail in the interest of time, because I was trying to show why an astronaut has to wear the gloves, which already Fatih has detailed it. First time ever for they were wearing the sterile gloves for MT1, thanks to Fatih, because he was instrumental in looking into what they are doing. So the astronaut actually touched the sample first without wearing the glove, and then he was actually helping them to, reminding them to use it. We came long way after collecting samples, so not only spore formers to the next generation. Anything in between. We have used some markers. We have done PCR, all these kind of things to measure the microbial contamination. So what are the processes? First thing is, in the DNA era, everybody wants to do the genome-based analysis, but will forget the traditional microbiology. But it should not, because of several reasons. One is you need the microbe in hand so that you can manipulate and know the functionality of it, and then get the benefit out of it. If you don't have that, only you have electronic data, digital data, that is not going to serve the purpose, but help. Students, please think, molecular biology is wonderful, but it is tool. Use the tool to understand the basics, okay? So you collect the samples here and then bring to the earth right now. Right now, it is still earth bringing to the earth. And then do all the things that you can do and then put it in a database. We are not forgetting this part. We are doing this also. But first, let me show you something about this. If you are thinking that biofilm is a problem, that is, where is the solution? You need the bacteria or fungi, right? This is Rhodotorula, a yeast. The red one that you are seeing are the yeast. The yellow one are the one that produces extracellular polysaccharide. This is the spacesuit material. Why I'm showing that? You don't have a laundry in space to do this spacesuit clean. We have some chemical, processing to clean. Astronaut A will use it, and then astronaut B, if it is the same size, will be pulling it, or she will be pulling it and use it. What that means? Leave behind something, right? So one of that is this. So if it forms like this, it will be a problem, right? So Ganesh was helping me to do this kind of things as well. So these are the one that is normal, but some of them are not normal. So these are the things that actually produce this extracellular polysaccharide, the spiky stuff. I said that we need a solution, right? Antimicrobial coating is one of the solutions, right? So this top panel is no coating, bottom is coating. This, when it comes in contact with the microbe, then that actually kills. Leave behind things. It kills. It's not cleaning, it kills. Okay. So this one is this one is Pseudomonas aeruginosa. This is Rhodotorula mucilaginosa, which is an yeast. This particular antimicrobial thing is okay to kill the microbe, bacteria, but not the fungi. So to do this kind of things, you need to have the microbe. That's what I'm trying to say. Don't forget the traditional microbiome. Oh. This is, previous one was stainless steel. This one is Teflon, because we need to test different materials to see whether it's working for different materials. Yes, it works for Pseudomonas aeruginosa. The top one colonizing it, the down one or two you are seeing it, but it is fully killing. Same thing here, Rhodotorula, not touching it. Oh. Now infernal, which is another one, same thing happening. And then this one is titanium, same thing. So that means there is no one size to fit all. You have to think about what is good for whatever it is. So this actually tried to attach onto the surface on top of it, which is not touching the antimicrobial thing, survive. But Pseudomonas is not this big. That's why it gets killed. But the surface area of the yeast and fungi are different. Okay, now we collected so many microbes. Then we have wealth of information from various different, funded activity by the research and development organization, as well as the mission since 1975 to now. We have these microbes in hand, and we were characterizing them. And some of them are novel and new, as I said before. We are not the bacteria which is new. Is it doing something? Is it beneficial? Is it problematic? Then you go in detail and do the taxonomy. Otherwise, NASA is an application-oriented place. It's not academic place. That's why I'm working with a lot of academic institutions who are interested in that, and then to describe them. One of them is this. So this I actually worked with the You're hearing? Hearing? Okay. I'm working with MTech. I don't know why it is like this. Go. When I'm hearing, what should I do? It's very here. Okay. It is and we named after Abdul Kalam, this one, because it produced a very nice, I named after Abdul Kalam, and it is actually producing a nice exosporium. This is actually carried by Ganesh, who is sitting. Why this is important? This exosporium you can peel off, still it will be okay. It is not going to die. But that exosporium, it uses as a cover and like a blanket against radiation. So this actually you can purify and use it as sunscreen. You can use it on several things, visors, like the helmet with the visors that the space station, astronauts are using in their spacesuit, everything that you can use that way. There are applications there. And the other one is, normally when you are going and looking for growing the yeast or fungi, you use some bactericidal materials like chloramphenicol. But some microbes are clever, some bacteria are clever. They grow on chloramphenicol, what they are. So this is also producing biofilm. And I named after, the person who sequenced first in space, Kate Rubin. So it's not only bacteria producing this kind of biofilm. Some fungi does. Okay, and these are this conidial spore, and these are the white thing that you are seeing are the one that actually extracellular polysaccharide. Okay, these are hyphae. This one we named as Pharangonchium torokii, and during that time I met this guy Marcus, who is also sitting in the thing. He's the fungal geneticist, and we named after Thomas Torok. He is the one who has, characterized the fungi that were black fungi that were grown after the Chernobyl accident. The Chernobyl accident is nothing but the nuclear accident in 1980s. And he's going to talk later tomorrow. So I gave you an example of spore-forming bacteria. I gave you an example of non-spore-forming bacteria. I gave you an example of fungi. Now it is yeast. Anybody who are doing yeast genetics or yeast biology, the Titan-like cells, that means by itself, Titan means bigger. When it is bigger, when it enters into your system, the yeast becoming bigger, the biggest one is Streptococcus neoformans, which is the deadly yeast, become bigger and produce virulence. So we found this because microgravity is simulating this, stimulating this. Simulated microgravity is stimulating the formation of. So we thought it is interesting. See this, this is normal gravity. This is with 5% CO2, ballooned under stimulated microgravity. What it is doing? What are the metabolomes and metabolites that it produced to make it like this? What are the lipids that are making? So we are working with a few people in India, Indian Institute of Technology, Gandhinagar as well. Okay, you saw all these things. Now, all the samples are coming down, and then sequencing as well as other things that's happening. So can we do the sequencing in space, which already NASA has shown with the MinION that they can do it. But can you collect samples and then do the processing automatically without, astronauts' time, without screw time? Let's look at this video. Please, hearing here, let me put this here. For the past couple of decades, microbial monitoring is happening based on omics technology.
Omics includes everything inside the microorganism. not just their species identification through their DNA, but also what enzymes they're producing, what proteins they're producing, and what metabolics they're doing. And as we study microbes in space, these are key things to take into account because those can give us clues and hints as to how microbes are changing and adapting to the space environment.
Able to process samples.
It is going to work in space because fluids behave differently under microgravity.
The SPI device can successfully extract DNA on the Earth's surface. However, the fluid properties of liquids in microgravity environments require engineering approaches, specifically the SPI to function in microgravity.
So if you don't design for microgravity, you can have fluids, you can have liquid on the top, you can have it on the side, you can have it on the bottom, you can have droplets ejecting, you can have geysers created, you can have all sorts of things where you really just don't understand what your fluid's doing or where it's going, and it can be very dynamic. And in this project, what we're doing is we are taking a closer look at the well plate design. We're modifying that so that it works predictably and efficiently in a microgravity environment. System that's designed for microgravity, the fluid at the base of the container, and like you're in 1G, it'll seem like gravity is actually present because all of it will go to this well at the bottom. And so you create a predictable, simple system that's passive and is self-correcting.
JPL and collaborators develop a pipeline, a microgravity independent pipeline, starting at the species, extracting their DNA, prepping the DNA, make it ready for the sequencing step, sequencing it on the biological sequencer, the Oxford Nanopore Medium, and actually be able to analyze the data on site, on the space station.
The data that we receive from the MinION can then be sent to various bioinformatics pipelines.
We take those files and process it as per the codes and open source software that are available to us. We do a quality check analysis. Each and every sample is checked whether we got the right results. We check it through the rare fraction charts, and the rare fraction chart tells you what type of assembly you will get and what type of organism or diversity you will get out of it. Once we have the quality reads with us, we do the identification and function analysis. The function analysis tells us what the system itself is doing here, which tells us what type of organisms are there in the sample or what type of processes they are doing.
Now that we have a pipeline put in place where we have DNA extraction on the SPI, sequencing on the MinION, and bioinformatic analysis, we had to put this pipeline to test. We decided to test this in a variety of environments that fluctuate from low biomass here at JPL at a clean room and high biomass in Yellowstone National Park on multiple biofilms that form in hot springs.
The hot springs of Yellowstone National Park provide an ideal environment for studying microbial ecology because they're remote, they have high biomass, and they are extreme environments. And this is analogous to how the SPI device will work on the ISS. It's a remote environment as well.
And what's so great about this test we performed at Yellowstone is we started getting data generated while still in the park. And before we even left the exit, we started getting sequence calls.
The data we generated in the field from the samples we collected there looks very similar to similar samples that were processed in a laboratory environment.
with the caveat that we were receiving our data almost real time versus days to months later.
This demonstrates that the SPI mini-ion bioinformatic pipeline we've developed here functions in remote settings.
So with this miniaturization technology, we will be able to analyze microbial population dynamics in space station. Perhaps even in a future crew vehicle that would go beyond the moon and Mars. So that is by our postdocs and students who actually helped me to think the process, and one of the inventor is a small business innovator company. And what I'm trying to say is, think differently so that you can get. So biggest issue there is nothing but pipetting, which is so easy, right? So we developed this and tested in the clean room samples as well as field, right? So and validated. And the next one is it going to work under microgravity? Without all this testing, someone was asking that, you do these things and, it takes 5 years to 15 years to mature and then, work in the space station, because they want to check each and every thing. They want to cross all the T's and then dots in the I's, right? So whether it is going to work in there or not. Let us take you to the parabolic flight testing. What does that mean? Because you collect the samples and then before coming down to the earth, it takes weeks and months. And even after coming to the earth, with all the things that you have, it takes around 4 to 6 months. Things will change. Microbes will behave differently if you are, preserving, right? So for that, we propose that, you take this to the space station and then with all the other things, try to sequence in there, because you have this min-ion, you have this right now tested. And then, we don't have this yet, but we are under development. And then we can get it within 9, working hours. So what is parabolos? You get weightlessness 20 seconds if you take this route. So around 1.5 hours to 2 hours time period, you will get 40 times of this 20 seconds. And then you can test some of the things, not all the things continuously, but you can test some of those stuff. Okay? So under these conditions, you will be doing some yoga. Okay? Let's tell me why we have to do this. I'm going to show you something here. Okay? See, he is pipetting. Still it is not flying. Now you started getting the microgravity, all the paper is flying out. Right? So now is normal, and then again another 20 seconds, all those things flying and then fall down. So this has to be contained. Just to visualizing the issues of the problems. So we took this in a 0G container. This is our instrument, after going through all the things, it can be, bolted. And then he's inserting this, he was my postdoc, he was inserting these cartridges before the operations, okay? So what operations are? So every 20 seconds you feel that way. Just like a monkey's up and down, okay? So whatever you are seeing, that is the time they are falling up and down. They feel the weightlessness. Okay. Now let's see this. Here is where he's sitting, our instrument. It is not only for our flight. It's going to be mixed with a few other experiments also. We actually got it free. Otherwise we had to pay. But they said, you have a space. Corona time, nobody wants to come. Do you want to go? Yes, let's go. That's what happened. Now, he is doing, this is experienced person. All of them are amateur. See how he is trying to fly. All of them are falling. Not everybody. Anyway, during the time, he is trapped. This guy is trapped. So he's looking at everything and whether it is going through or not. So, okay. So that's what it is. Once you mature at a various different things, then now we are flying that to space station. It is flying February 2023. Okay. Next one is environmental microbiome. Okay, isolation of extreme microbes from deep sea. I don't need to explain you this group that microbes are everywhere. You need to look for it, but it may be a different kind of microbes, right? Antarctica, around 2.2 miles down, there is a brine solution and the water, seawater is flowing around. So microbes were isolated from there, but we need a technology to clean the cores that going through it. Otherwise, you do not know which one you are going to look for it, right? And then likewise, we went to the Rio Tinto, which is in Spain. It is the Red River. That is because of the iron ore that is flowing through these waterways. And then we developed this, sample collection technology, and we took it to the field and collected using those, metal filters, which are compatible to the high acidic, water. And then not only ours, so many other instruments went and then got this stuff published. And then this is hyperthermoacidophilic, both temperature as well as acidic. See this one 2.9 pH, 64 degree, 100 degree, 3.8. So things like that. We collected these samples and used the phylochip DNA microarray and published about our thing. Why do we need to do all these things? We need to know our backyard and then learn from there and then use it for space related work. That's why. Here I'm not going to go through because of the issues. The other one is very interesting is Nick also involved in this thing is 2,141 meter subsurface of a gold mine, molybdenum mine. Okay, we found a microbe that is found in the 2,000 meter down, same in our spacecraft assembly facility. What does that mean? So we have to look for what we are interested in. Okay, if this is a spore former, okay, 2,141 degree, and also we were getting it on this, Phoenix lander assembly facility, exact same. We published that, and then we went to caves. So these structures that are formed also microbially mediated. Why I should you go to the cave? Because the microbial diversity of the clean room is mimicking the unexplored caves. We are going to the other planet and exploring things, right? Not explored yet. So we have to build appropriate database and then say, hey, these are the one hitchhiked with you. And then you took it along with you and you are saying, hooray, I found something. Eureka moment has to be appropriately managed. So we published this thing also. The orange color thing that you're seeing are the novel microbes. Okay, belong to certain classes that is first time identified. Now go to the deep sea to 2,500 meter down. It is actually went from South Africa to the Australia around 3,000 miles on the around 35 days expedition. Okay, so using the submersible because you cannot dive down to the 2,500 meter. You will not come back. Anyway, before going down, you are giving your life. You write off your life saying that you are giving your life for the science. Otherwise, they don't take you down. And I'm a proud Tamilian who went down there first Tamilian who went down to the 2,500 meters. This is how it is getting operated. Okay. So this submersible from the mothership. They put you down there, okay, for about 8 hours to 9 hours. You will be inside, and then you go through the places and then collect samples and come back. And this is called Kali chimney. You also collect by putting some sort of instruments in there where this 420 degree, hot samples come out. So at 420 degree, the hydrothermal vent is clean and clear. Immediately when it comes to cold water, precipitation happens. Iron, sulfide, all those things happen. That's why you are seeing the mount. Here, some of the shrimps and other things. So we insert some sampling there so that the hydrothermal vent fluid is going through. It is a ceramic, so it collects something. So those are the things, because we want to know where life exists. So Derek Lowley's lab is telling 121 degree, life is multiplying, but he's not giving the samples to anybody else. But Caltech proved 110, it is multiplying, it is Pyrobacillum aerophyllum. You can get this archaeon and then test it by yourself. So this is the limit that we know today. That's very important. We know today. In science, you cannot say no, but you can say, I don't know. Because next generation technology will tell you, you are getting better. Indeed, NASA's terminal sterilization based on the temperature that before this was found. Now their concept is changing. Let's push it to 125. Let's push it to 250 kind of thing. So in this range, because from here to here, you have 300 plus, this particular vent fluid that we took, this is real data. Okay, in this industrial thing, that's where you have the thermoflax, the plume comes in, whether it is whether you are able to find the microbes or not. So we need to devise a new sample collection technology here. As you know, 100 degree and above, all the kind of material will melt. So you need to have a metal filter. During that time, we didn't have a metal filter of the size 0.1 micron. So we specially ordered those things and then sequentially filter, because you do not know the precipitate comes and then clog your system, right? You insert your probe inside, pull the water through that nozzle. While you are pulling the things, that you are also measuring the temperature. What kind of fluid is coming through, whether it is getting contaminated with the 4 degree nearby water or not, and then pull those things and then sequentially filter at different micron, 90 micron, 60 micron, 7 micron, 0.2 micron kind of. And then we tested locally in the lab, various pressure testing, various dive testing, all these things. Then we had a chance to, this is the, you can look into the various details of it, but this is where we took it initially, a shallow place, whether it is working or not. We deployed it there. There is it's not a very hot thermal area. It is only 65 degree. So we went and then collected samples to test how the things are working. Then we had a chance to go down to the, remotely operated vehicle. We call them as ROV, and then mount it in this and then collect the samples. And the samples collected, brought back, this is the mount, this is how it is done. And then the temperature that we have collected this is 280 degree on this 10 minutes period, which took around 10 liters. Okay, we have this data thing. Now you can analyze here written, sample collected at various depths, and then vent temperature up to 313 degrees centigrade. And the problem sometimes you happen, something broken, And then we were able to, rectify that. And we collected. The answer is either life exists at 280 degrees. The answer is no. I should not have said no. It is not known to the level that we right now, to the technology we know today. Okay. Okay. So applications. Kalamiala story I'm going to show you. So repeatedly, this genomic-based analysis, metagenome-based analysis, we started getting some signature that is not known, that is not described yet. 5 minutes? Okay. So we published that based on that, named after Kalam. This is a novel genus, Kalamiella. Previously, species. Okay. I said it is application, right? After some time, Asif is sitting here, his boss, Rekha, actually first report this as a pathogenic bacterium for the uropathogen. Uropathogen normally, do the urease activity and other things, but this particular thing is negative for urease. It's a different metabolic pathway. It forms this kind of stone, kidney stones. Okay. After that, in 2022, May, I think, I got a call from New York, hospital. They found Kalamiella from a patient who has a 56-year-old woman with a medical history, but they need to hospitalized in an emergency, and then 14 days of intravenous antibiotics, allowed them to treat it. And then I got another collaborator, sent an email saying, hey, we got oral microbiome has Kalamiella. How the hell that space station thing is in here? And then he's working on that now. How different all these strains of Kalamiella. And just before coming to India, this was the latest one from Germany. They found one. So we got all these genomes, all these strains. We are trying to see how this Kalamiella story is, confounding. The Methylobacterium story, and I named after one of my senior, Ajmal Khan. And this is a rod-shaped bacteria, which Nitin has pointed out a bit. This produces hormones that helps the plants to grow better. We know only genomically right now. The validation needs to be done. Some of the metabolomes of this will help us to understand what metabolites actually allow the plants to grow better. So I named after my very well good friend, and you said 5 minutes. It's 12:51. Is it right? Then I have to stop here somewhere. Computational, I'll do this. Klebsiella story. Then I will end it here. So persistence and spread. So if you have some of the problematic microbes, as I said, you need to find some solution, right? So here, Klebsiella this is the flight 1, flight 2, and flight 3. Flight 1 and flight 2 are 3 months apart, and flight 2 and flight 3 1 year apart. It is not intentional because of the SpaceX 6 blown away. We did not have a flight to go and collect samples. So flight 2 and flight 3 had 1 year. And then locations that Fatih has already shown you, location 1 through location 8 here. Now we started getting some pattern of the Klebsiella, which is a BSL-2 microbes, and then persisting the flights and also spreading to the other locations. And also another one, this location 2 is nothing but the toilet, space toilet. So that is not, it is also there here, but in a lower abundance, and other things, supersede. But basically, what we are trying to say here is the spread of Kalamiella species, which is an opportunistic pathogen coming from one strain. If so, how to find it? And then can we do it from the metagenome analysis? We have the data in the gene lab. Is this from one single strain or not? If so, how to eradicate? And then phage therapy. There is a poster right now that Anup is and his fronts, the students from Indian Institute of Science are having there. You can talk more there next to it about the phage therapy. But this is what the collaboration with Karthik's group on the metabolic modeling. So we have given all the metagenomic data, request them to predict the possible interaction between the microbes in the community. So he come up with this saying that there is a microbial warfare going on. Some of the things are showing the parasitism, some of them are ammensialism. That means this Klebsiella, if you take it, this is actually killing most of the fungi, but some of the yeast actually killing here. That means this produce anti-fungal compounds and Rhodotorula producing antibacterial compounds. Let's look at into the fact, right? You predict that, but you have to experimentally prove. This one is a Aspergillus fumigatus only under normal gravity. It's beautiful flower-like structure with the stalk, which is hyphae, and then conidial spores here. If you mix with Klebsiella pneumoniae, it's red that you are seeing here, actually grazing those spores. Now, if you take the Aspergillus and then do the simulated microgravity, it is actually, to some extent, is weak. That's what immunocompromised state is. If it is immunocompromised state, the Aspergillus fumigatus and Klebsiella double punch, actually balding the whole thing. So what did we talk today? Dream big, believe in you. Anything is possible with process. There is a teamwork. Okay, appreciate your competitors who are achievers so that you will be having more things get done. Winning is the end product, but a process is necessary to achieve. L3 formula, love yourself first and eventually others. Learn from others' mistake. Everybody intelligent, but smartness is to learn from others' mistake. Listen to winners. Follow what your heart says. So this is because of this team that we were able to do it. Still, we are hungry. There are so many things to be done. That's where we end.
Hello, is it working? Yeah. So thank you so much, sir, for this enlightening talk. So I have few questions, like you have collected the sample from extreme environments, and you get to know with the metagenomics, like which species are they. So, but I just wanted to know that how difficult it is to culture them, like to know like more about them. It's like you are saying, it is not just we just wanted to find what are they, but actually we wanted to know what they are doing. So just looking at their genome, it's just we know that, like, yeah.
Yes. So very good question. So that's what the metagenome phenomenon is. That's what we are saying. From the metagenome, if you are getting again has some functional properties based on metagenome you are seeing, then you have to devise things appropriately. Is it barophilic? Is it require a pressure with the temperature? All those kind of cultural conditions you have to develop to get it done. It is not easy, but it can be done with appropriate cultural conditions and other things. But some of them you can never, because symbiotic microbes and other things, you're never able to do it. So then you can close the genome like MAGs, metagenome, assemble the genome, that are not culturable. From there, you come to know what are the conditions required for them so that you can do it in the lab. But we are succeeded in only 1 to 10% of them to regrow them. So still a long way to go.
Okay. Next question is, it's very basic. Like as you were saying that spacesuit, if one person is using that and second also is using that. So there is something which the first person is leaving behind, right? So we call it like a biofilm or something is there. So do we every time go and check, okay, is there anything, or just next person comes and use that?
No, that's a process. That's why process. So you measure the microbial diversity over a period of time with a different number of spacesuits, and you come to know what other things are there. Is it problematic? That's very important. Is it problematic, or your immune system is actually fighting against it or not? So if it is problematic, you have to come up with a solution like antimicrobial coating or some sort of chemical in nature that can eradicate, kill them.
Thank you, sir.
Yeah.
Hello, sir. Thank you for the wonderful and insightful presentation. I just have a quick couple of questions. The first one is much similar to what you had asked, so maybe I'll just tweak it a bit. Any attempt towards a development of in situ samplers for collecting your samples from Mars or any spatial locations? Because considering the microgravity or the pressure and the temperature that exists there, I'm talking about cultivation of microbes after getting the samples on Earth. Any attempts or any lights on those and in situ samplers?
Did you miss my video? Did
No,
it come later or what?
No, sir, I was
that-
there. Just had a doubt about the maintenance, the pressure and the temperature. So,
Okay, you asked multiple things. So how do you collect samples? We have technology right now how to collect them, but we don't have a launch pad in Mars. That means we are still working on the engineering technology, how to bring that sample back from there to Earth without getting contaminated by either the transit material, or the Martian material should not contaminate our Mother Earth as well. So that technology is being developed as we speak. If you simply sit, the time will go by. Now we have the technology how to collect and then keep it intact. That's what the video I showed you. Yeah. The other one is, how do you keep the Martian temperature and atmosphere during the transit to come here? First of all, you need to know how to transit. So maybe the Martian orbiter will go from there, like a Martian helicopter that we showed how to do it. Like from the orbiter, something will go and fetch the sample cache that we left it there. We know exactly which position there. So it can fetch and come back in the orbiter and come back to that without a launch pad in Mars. Okay, that's one way of engineering to get the samples back. After getting the samples back, what to do, that already Dr. Bernardini explained you. You were there in the beginning, right?
yes, sir.
So we still has to build BSL level 5 kind of thing to keep it safe. That will not go to the Mother Earth and then eradicate everything. We do not, because we are dealing with something unknown, right?
Yes.
So all these things happening, a lot of unknowns, but we know how to collect samples.
Okay. So I think in the interest of time, we'll have one last question from Daniela. So have there been any early experiments on the ISS? Adaptive laboratory.
I don't think so. So far in space station, I think somebody else also asked about evolution. People get confused evolution with adaptation. It is only 22 years old, the space station. Things should not evolve there. It will adapt. Two different questions, okay? So theoretically, it takes millions of years to become a new species, theoretically speaking, okay? The rest of the point mutation and other things, phenotypically different, which is not evolution. So this is the biggest topic that we always discuss with the people. People should not think 22 years of space station will make a novel species. So adaptive laboratory evolution, I don't know how long it will take to make one species into the other species. That is not happening.
Okay, I think there are a lot more questions, but we will take them during the lunch break. Thanks again, Venkat, for a wonderful talk, and thanks to all the speakers for the end of the entire session, for actually sticking to time perfectly, and of course, giving very exciting. So we have a small token of appreciation for all the speakers. So let me start with Venkat.
Now I request Dr. Venkat to give away the mementos. Yeah. So now it's Dr. David Reed. And Dr. Fatih. I would like to once again thank all the speakers and the chair for this awesome session. So now we will be departing for lunch. I request all the poster presenters to put up your posters in the hall. It is just towards your right, and when you get out, it will be towards your right. So there are poster boards with your numbers, and the numbers are there in the document shared in the WhatsApp group. You can also check in with us, and please display your posters accordingly, and you can go back to lunch. We'll be assembling here at 2:30 p.m.
So one other thing for the students who have the posters, try to have early lunch and then be there at least, 15, 30 minutes so that the people will come and talk to you. Simply leaving the posters and go, it's not good. So that everybody can walk and then talk to you. Okay.
Nitin, are you there? Nitin?
Yeah, I'm here. I'm here.
Oh, you are? Okay, boy, that took a long time. Is everything all right?
2 a.m. here. And the whole committee can hear us.
I don't understand this.
Okay.
I only have one person online, so I don't know if there's anybody else on the other end, and I just want to confirm that.
Yeah, there are 100 people on the other end. This office of GEIIT Madras, that's 100 people literally sitting in the room.
started to me. So for some reason, the first speaker is. Okay, we'll make you first. Already there. You're already there. I'm thinking. Alex, go ahead.
Welcome back everyone to the fourth IBSE Symposium on microbiome in space, environment and in human health. For the second session on environmental microbiome, we have Dr. Alexandra Rosado as a chairperson. Dr. Alexandra Rosado is an environmental microbiologist specialized in basic and applied microbiology, microbiome sciences, and microbiology of extreme environments. He is also a professor of bioscience at Biological and Environmental Sciences and Engineering Division, Christ Kingdom of Saudi Arabia. He is a former professor at the Federal University of Rio de Janeiro, and he is a former director of Institute of Microbiology. He is an associate editor for the Journal of Microbiological Methods, Frontiers in Microbiology. We welcome you.
Well, thank you so much for this nice introduction, and it's my pleasure and honor to be here sharing the second section of the day. We have some changes in the schedule, and I know a new change in the schedule, but it will be exciting. I think it will be fitting perfect with the session that is about environmental microbiomes and extreme environments. So first, I'd like to introduce our first speaker, Dr. Scott Tighe. Unfortunately, he was not able to join us face to face, but Scott is the technical director and the lead scientist at the University of Vermont Advanced Genomics Lab. He has expertise in areas of DNA sequencing, including messenger sequencing, next generation sequencing, microbiome sciences, and metagenomics. As a group leader of the Extreme Microbiome Project, that's actually a very important project for those that are willing to work on extreme environments. His research focuses on characterizing and detecting microbial life collected from extremophilic and unique sites from around the world. He has developed new methods and techniques for DNA extraction, for exoplanetary and advanced microbial devices, which could be deployable in a spacecraft. He assumes various roles and the chair of the Metagenomics Research Group and board member of the Genomics Standards Consortium are a few among of them. So he has several different roles. And please, Scott, are you there?
Yep, I sure am. Can you hear me?
Yes, perfect.
Okay.
So
Thank
Scott,
you.
we'll follow the same flow, like 25 minutes and 5 minutes for questions, okay? And I tell you,
Absolutely.
we have 5 minutes left. Thank you.
Okay. Well, thank you for the wonderful introduction. So I hope you can all hear me. I'm very appreciative for the invitation to speak here, and I'm sorry I couldn't be there. I know it's a wonderful event and a lot of work put in by the organizers. So my name is Scott Tighe from the University of Vermont. I want to talk to you about strategies for profiling the dark matter or the unknowns that we see in extreme environments. So just need to launch my PowerPoint here. One moment. Okay, so I already had a wonderful background introduction here, but I have been doing I do want to just say the first microbiome project I did was in 1985. So that kind of gives away my age. And I do run 4 international or participate in 4 international consortia, and I really want to focus on the Extreme Microbiome Project. I also have one NASA ISS grant that I've gotten, and we're going to launch the MicroTitan device to the International Space Station. I'll talk a little bit about that I've been working with Venkat on for a number of years. So the reason that
one second. Can you make it a presentation mode so that we can see bigger?
Oh, I'm going to jump over to, I think I'll jump over to a PowerPoint instead of is this?
Okay, got you. Okay.
Is that okay? Venkat, is that okay?
Yeah, it's okay.
Okay, sorry, I lost your audio. So the reason that we work in extreme and novel environments is a couple of fold. One is we want to describe new organisms, of course, that's the dark matter. And we want to discover new biosynthetic gene clusters that may in fact have some commercial values or some clinical value. And we can also use this to discover new biological pathways in the environment and help discover the origins of life, truthfully, from a planetary evolution standpoint. But I have to say, it's also quite fun for those of you that are doing hardcore research. You know, when you do extreme files, it's quite fun to see new things and discover new things. So it's also a proving ground for new techniques. So when we develop new lab techniques, analytical techniques, these are the hardest things to extract on the face of the earth. That is for sure. And we also have great collaborations among all the disciplines. So we have geochemists and microbiologists and bioinformaticists working together. So we know that extreme environments are non-standard in temperature and pressure, from flames and hypersaline to deep oceans. So it does make the microbes very difficult to crack open. So routine methods are not optimal. So this is where we use special enzyme and freeze thaw and beater beads. And library preparation techniques for low input are also a problem. And all of these require special handling and concentration steps that I'll talk about in just a moment. So it's a battle between how do you collect a sample, how do you preserve a sample, how do you lyse a sample, and most important, the controls that you have to run to maintain the proper quality control of the data, because it can get away from you quite quickly. So we spend a lot of time working on trying to develop DNA-free reagents, and I will show you what we've been doing over the past 5 or 10 years. So the Extreme Microbiome Project has a lot of facets to it, and we do a multi-pronged strategy. So we actually do microscopy, we actually do DNA, RNA extractions, and we do culturing. So all of these have to work in concert with each other. We don't just run DNA extractions through a nanopore. We don't run DNA extracts just through a HiSeq to get taxonomy or biosynthetic gene clusters. We try to match it up with the culturing and the microscopy, because seeing is believing. So this is the theory of minimum truth, if you will, where if you look at something under a microscope, you might not see it in metagenomics data, and you might not see it from culturing. We see this all the time, and I'll show you an example where you'll culture something, and what you get on the culture results do not come out in the metagenomic results. Likewise, a lot of things in microscopy do not come out in metagenomic results because you can't lyse them. For example, some spores, coccidians, diatoms that aren't in the databases, nematodes, and the stuff that's you can culture that doesn't show up in metagenomics is they're so hardy, their cell walls are almost impervious to crack. So currently we have 14 sites around the world that we've indexed, and I'll just show you a couple of nice pictures of it. This is Lake Magic in Australia. This is a pH of 1.5. These cells, you can imagine, are quite hardy. The Dead Sea, Lake Hillier in Australia. This is a hypersaline lake in the southern part of Australia, in the archipelago. We just published this last week. The Ethiopian toxic hot springs. This is probably the worst of them all because it's hypersaline, sulfur, and high temperature, and most difficult to get to for a number of reasons. Deep ocean brine lakes are lakes that are under, sorry? The deep ocean brine lakes are actually lakes that form on the bottom of the ocean. And this is Mandy Joy that are collecting samples in the Alvin Sub. And this is at 3,500 meters. And you can see she's at the shore of an actual pond that's at the bottom of the ocean. And these are hypersaline lakes. Mobile Cave is another site. This is Sherban, who's collecting new insects. Also many new bacteria we've described from this. And you would call this dark matter for a couple of reasons. One, it's a very dark cave. And also the organisms that we get from there are certainly undescribed. The deep ocean sediments in Greenland, we've done a lot with. These are also in the 2,500 meter range and quite unique. And Antarctica dry valleys, we've done a lot there with the paleomats and the biofilms that are located about a foot or about a half a meter below the surface in the dry valleys. And this is one of my great slides because one of our speakers that you all know, Stefan Green, this is the Dorsta Hill crater in Turkmenistan. This is Stefan Green dangling over this burning crater, so bravely going to collect samples for the Extreme Microbiome Project. Yellowstone Hot Springs, this is a project we've been working on with Venkat at Jet Propulsion Lab, and then microbes from the ISS we've done with Venkat. And then most recently, our project that's going to the ISS, and this is an automated DNA extraction platform that was pioneered by Venkat about 5 years ago. And it has a hermetically sealed zero gravity cartridge that has DNA reagents in here that astronauts can use without contacting it directly. And you inject a sample into one of these injection ports, and it automatically through this instrument, which can also be battery operated in the field, will extract the DNA that will come out the other side, and you will pipette it out the other side into a library prep for Oxford Nanopore. And this is scheduled to launch in February. So we're scrambling to get all of our stuff done, and we're going to extract DNA, microbial cells, yeast, and we're going to test 3 microbial preservatives. So that's one of the things that we worry about in extreme microbiome projects is we have sample collection issues. We have to preserve samples. Of course, DNA, RNA extraction, library synthesis, data analysis are all issues with going after dark matter unknowns. I'll show you some examples of what we've gotten in the past. Sample collection isn't standard. I think this goes without saying, but it requires special people, special equipment. And I say spacecraft because that's what we're doing with MicroTitan. But again, we have a Alvin Sub here at 3,500 meters collecting samples. So we want to make sure our samples are prepared correctly. So having this correct sampling tool, not just autoclaved, but flame sterilized, incinerated, sterilized, and using disposable DNA-free, if we can, reagents before we go and collect these samples because we don't want to contaminate them. Depending on the environment, you're going to do inline filtration or some type of filtration. And here's a picture of the older millipore filters that we used to use, and then we graduated to the SteriVec filters, but SteriVec filters don't work in Yellowstone Hot Springs or the Dankill Depression because they melt, so you have to go to these stainless steel filters. Or you can collect grad samples and use an ANOVA prep concentrator tip such as this, where you can concentrate liters and liters of waters to microliters. Another technique we use is gradient flotation, where you take something such as percol or zinc selenide and actually float a sample underneath it to do a gradient flotation, and this separates debris, high density debris, from biologicals. And we do this routinely on the Greenland sediment samples to get biologicals away from dirt, if you will. Preservation is, we can't always do fresh frozen. We'd like to do fresh frozen. It really is the correct way to do it. But when you're out in the field, you have a multiple way of doing it. You can use ethanol, DNA, RNA shield, LifeGuard, or some of the new stuff from Rapid Labs like the FXV. The thing about DNA RNA shield, we tend not to use it in our lab because it actually causes lysis to the cells. So if you want to spin down your cell population, some of them would be lysed and you'd wash away some of the supernatant in the DNA, whereas ethanol actually preserves the ultrastructure of the cell and prevents leakage. So that's what we go to, a 90% ethanol PBS, and that preserves structures and the DNA or RNA, and I will show you a couple of examples. And we do not use RNA later because RNA later is 7 molar ammonia sulfate, and that's got a buoyant density of 1.22. So it itself is a gradient flotation, so you can't spin down samples, which is a problem if you want to wash them before an enzymatic reaction. So we know that all preservatives not creating equal. Depending on what preservatives you use, you're going to change what you recover taxonomically or your diversity. So each preservative you have to consider for your environment. So a ethanol preservative might not work in a hypersaline situation, and then a DNA RNA shield might not work in a high temperature environment. These are things that need to be considered for these types of environments when you're going after undescribed organisms in extreme environments. So here's some data from ethanol that we tend to use. This is on the top left. This is RNA that was recovered from a microbiome samples. This is 3 years old of a sample that was recovered in Greenland, and we just had it stored in the refrigerator at 4 degrees centigrade for 3 years. And you can see the RNA is perfectly intact. And the DNA has ultra high molecular weight. So if you want to do PacBio or nanopore sequencing, the DNA is intact. On the right-hand side, you see a panel of the cell permeability. This is important when you preserve an organism. You don't want the DNA or RNA to leak out into the supernatant. And this is a leak down study where we prove that ethanol keeps the RNA and DNA intact into the cells and it doesn't leak out. And that's after 2 years. And it also preserves the ultrastructure. These are just a couple of images of E. coli and Enterococcus where we grand stain them after sitting in 2 years of ethanol. And as you know, E. coli is gram negative, so that comes up as pink, and Enterococcus is gram positive purple. It also allows you to do enzymatic digestion of cell walls, because that is incredibly important with extreme oils. If you want to get the DNA out of these, you got to get rid of the cell wall. If you preserve them in such a way that you make the environment not conducive to have an enzyme work, such as metapolyzyme or lysozyme, you eliminate that from your DNA extraction approach, and that's a very bad idea. So it's nice to have a way to wash something off your cells and allow them to still be a substrate for an enzyme if you so choose. Nonetheless, DNA extractions are the biggest variable in all this. We know that when you look at, these are 2 examples up here in the upper right from Antarctica biofilms that we've collected. You could take a paleomat from a lake, Lake Vanda, or a modern mat that we got from Bonnie, and we use 3 different extraction protocols, and the microbial yield from each DNA extraction is different, which is not a surprise, but they also switch. So a mobile or a DNA power soil performs differently on a modern mat versus an ancient mat. And that's important to know. And that was highlighted in a 2012-13 study. Stefan Green was part of this study where we took a number of organisms and we tried 9 different DNA kits, and we see you get different extraction efficiencies for different organism types depending on the DNA extraction kit. So these are things that we're trying to work out in the International Metagenomics Microbiome Standards Alliance group to see what way we can standardize DNA extraction and actually bring DNA extractions further along. But our approach to date is, I'm just looking at this flow chart here, and this is outlined in this publication here, where we actually take a sediment and we actually will heat it to 70 degrees and knock out the DNases. Then we add sodium azide, and then proteinase K. The azide is to prevent microbial growth when we do the next step, and the proteinase K is to help digest biofilms. And after this step, we actually heat the sample to 80 degrees to deactivate the proteinase K, and this is now a thermolabile proteinase K from NEB that's now available. And then we add either metapolyzyme or lysozyme and do this should say 24 hours, sorry. A 24-hour digestion of the cell wall. Once the cell wall is gone, we do a freeze thaw from -20 to 70 degrees and then go into our favorite DNA extraction kit. DNA beads don't actually lyse cells completely, nor do chemicals. Here's just an example of Micrococcus luteus, where we actually took Micrococcus gram-positive organism, very high GC, and you can see we exposed to the C-tab, phenol, SDS, C-tab with beater beads, C-tab with beater beads, and phenol. And all of these you have intact cells. This is no surprise. There's no DNA kits that actually get you all cells and all DNA from all cells. but cell wall digestion increases this. So you should use metapolyzyme or lysozyme if you can, in the situations where you can. And here's an example where you take cells, same one, this is Micrococcus luteus, we're exposed it to metapolyzyme, and you can see that we got rid of the cell wall. You can see that there's spheroplasting here, blebbing, and then if you just do isotonic shock to them, you lyse all the cells. So can't say enough about doing an enzymatic pre-digestion if you have the bandwidth and the situation for it. And this is you can see the cell walls have been removed from these cultures because the refractive index of this broth culture changes. And as of September 2022, there is a DNA-free metapolyzyme. This was actually something that got requested by Venkat a number of years ago, and it's finally released, and it's really for low input and space applications. This is a list of the enzymes that are actually in metapolyzyme. You can make it yourself, but I don't recommend it. I did it for years. And it can be applied to swabs if you so choose, and we have proven that you get higher yields with swabs if you want to treat it with an enzyme. The next thing to consider is biofilms are really a problem. And we're just starting getting into that. Every extreme environment is loaded with biofilms. This is a microbial mat from Lake Vanda. These microorganisms are completely protected by the biofilms. Same with Yellowstone. Likewise, bacterial capsules do the same thing. So you got to get to the cell before you can digest the cell wall. So the approach here is to digest the biofilm, then digest the cell wall. Once the cell wall is gone, then you can digest the membrane and recover the DNA. So really, it's a three-step process to do it right. And we're working on a multi-enzyme cocktail right now. Stay tuned for that. That'll probably be 2 years out, beta testing by the end of the year. Freeze-thaw cycling is important. And if you do freeze-thaw cycling, just remember this is one of the techniques we use to crack open oocysts and spores and pollen. And the technique behind this is the slower and the not so cold you go. If you go from -20 to 70 degrees, -20 will grow a very large ice crystal. However, if you do -80, which some people do in their freeze thaw, the ice crystals don't grow as big and you get much less lysis. So if you're going to do that, it's actually quite conducive to lab people because you can just put it in a freezer and walk away, a normal freezer and walk away, and you get fairly large ice crystals that can help crack open spores. And avoid -80 because that's not going to help you. That's more like a flash freezing approach. So a little bit on methods. I think all of us are using Illumina and nanopore sequencing. Some people are doing PacBio. We are now approaching metatranscriptomics as a regular microbiome assay. So Illumina methods using a sample that's ribosomal depleted, such as the NAB ribosomal RNA depletion for metagenomics. But we're also, and Stefan Green's participating in this, we're using a ZAP-R approach where we're actually, this is a low input for moderately degraded DNA. And this is really important to us because all the RNA that we get is moderately or severely degraded. And in a lot of our environments, we only end up with 10 nanograms, and sometimes we only end up with 3 nanograms. This is being worked on by Takara and the ABRF team. So we're excited about that. The problem with the standard RNA reduction protocol is you need 100 nanograms, and often you don't get that from these sites. I'm not going to say anything about 1618S because that's an area that Stefan Green is an expert on, and he's in your audience, and he is probably going to talk about that. But I do want to make some comments about ultra low input because half of our samples that we get from extreme environments are really low, down in the half a nanogram range. And I think that the JPL crew also sees this. And this requires some type of pre-amplification before you can get into, say, a nanopore situation. It is very sensitive, but it is very noisy. So the sequencing strategy is if you have a lot of DNA, you can go into a standard kappa RNA-seq into an Illumina. And likewise, nanopore, you can go into a ligation reaction or any of the reactions quite successfully. That's never a challenge. It gets a bit more challenging when you go lower. So nanopore, when you're down in the 1 to 10 nanogram range, we have to go to a rapid PCR approach. And we can't do native sequencing, but there are GC challenges when we get to this level because GC affects the rapid PCR barcoding. But of course, Illumina is fine. Nextera works very good. No problems there. It's only when you get to the 10 picogram and 200 picogram range where we got to do preamplification. We use MDA with a T7 endonuclease debranching strategy, and we've been working on that for over a year now. And we just ran it a couple days ago on an LSK114 and a nanopore. But of course, you get dropouts where the branching occurs, even if you're debranch. Sorry?
Scott, you have only 4 minutes now.
Yep, that's okay. I'm almost done. So here's some multiple displacement when you compare the two. And I know we want to take some questions if we can. So I'm going to just jump ahead onto a little bit that's quite important. Is we know the kitome is very important. Reagent contamination plagues us when we're dealing with ultra low input. We know that's as a problem. We see it all the time. So we work very hard to make sure we have DNA-free reagents. And here's an example of some background contamination from several different DNA kits. And I do want to make a comment that E. coli is not a sample contaminant. People say it's a contaminant. It's a bioinformatics problem. So keep the E. coli just doesn't hang out. So it is important to have positive controls and negative controls carried out through your whole process. I'm not going to say anything about data analysis. Data analysis is handled by Chris Mason's lab, but we do have data analysis for long and short read and biosynthetic gene clusters. But I do want to just take a couple more slides and show you some data. This was Lake Hillier. This was an ultra hypersaline lake. You can see that saline crystals that are in the lake in that image. And you can actually see when we looked at whole genome and the bacteria amplicon and then the eukaryotes that we are able to identify that Selenobacter, Psychroflexus, and Denaliella were the organisms. But I want to point out that the microscopy showed Denaliella and we actually sequenced it. We cultured Psychroflexus and we actually saw it in the DNA sequencing results. So that was a nice confirmation. And we did biosynthetic gene cluster analysis. We were able to identify 2 biosynthetic gene clusters from Lake Hillier. So in summary, I just want to say that this is really a collection and extraction and handling exercise when you work in these. And it's really important to understand the DNA extraction is the limiting process here. But these are extreme environments are a valuable source of biosynthetic gene clusters. And I encourage people to do microscopy and culturing and make sure DNA-free handling is engaged whenever you're in ultra low input environments, or else you're going to be dealing with a contamination nightmare. So many of you are on this list, and I have to say special thanks to the organizing team. I know how hard it is to put these international meetings together, and there's so many people involved in this. I'm not going to go through it. So I know that I'm running a little over time, so I'll stop there.
Thank you so much, Scott. It's nice talk, great talk. I think the sound is not very good yet. So I think we have time for some questions. Anyone from the audience would like to make one question? Anyone? Not really. So Scott, can you read the Slido? There's several questions there, but wow, I can go for one question. The first one, what is done in case of RNA extraction in stream file if we wanted to look for mRNA?
Oh, yeah, that's what we're doing from an mRNA, polyadenylated mRNA approach is what we do is we take the RNA and we go straight into a Pico V2 Takara kit, and it works fantastic.
Great.
Simple answer.
Yes, very good. Marcus?
Hi, Scott.
Hi.
Really nice talk. Yeah, we're missing you here, but anyway, I am very curious if you're testing these novel cocktails of enzymes for fungi.
Yes, great question. So Marcus, yes, so we have the metapolyzyme. We incorporated 2 fungal degrading enzymes, lidocase and chitinase. We haven't done any fungal degradation studies with them because of bandwidth, but we did include them, and that's all I can say there. But if anybody wants to try it with fungi, I can probably get you some aliquots of metapolyzyme to do such. We did originally have a massive, we're coming out with, we started a metapolyzyme 2.0 that was going to have far more components in it for fungi, and it was going to be a two-part system for plant, but we haven't gotten the bandwidth to do that yet.
Thank you, Scott.
Okay, thank you, Scott. Thank you very much for your nice presentation. And let's move to the next guest.
One second. So, Fumito-san, just try to start your presentation, share yours, so we know where you are, okay?
Can you hear me?
Okay, please be there like that. Now the chair is going to introduce you.
So it will be Fumito now, yeah? So our next guest is Dr. Fumito Murayama. So of course, be presenting virtually. Dr. Murayama is a professor in the section of Microbial Genomics and Ecology of Office of Academic Research and Industrial Government Collaboration, Hiroshima University, Japan. He's also an honorary professor at La Frontera University in Chile. And overall, his lab focuses on elucidating interactions between microbes, symbiotic hosts, and the environment. So please welcome Professor Murayama, and please, the stage is yours.
Okay, thank you very much for your kind introduction. Before I start, I'd like to thank you, Professor Venkat, giving me a chance to present in front of you. It's my great pleasure to have my recent and ongoing job in front of you. So today I'd like to talk about the microbes around us, especially the microbe in the Japanese built environments around us. So
please, can you put in a big screen, share the screen? The presentation. Yes, it's
right.
in there. Presentation mode, yeah.
Presentation mode?
In the bottom.
Okay, please wait.
Yes.
Oh.
No, the other way.
Okay. Okay. So, as you see, now we are spending our most of the time in our house, and our house is very different in the world, depending on country and environments and climates. But more importantly, as you know, our living environment is modern environment is just built maybe around 300 years ago. It means our modern environment, our built environment is kind of extreme environment, so to say. So all days we are living in this kind of nature houses, like a cave. And so it means this kind of modern extreme our environment is a very new environment for microorganisms. And this our environment most important characteristic is tight air tightness. So it means our environment is filling with microbes with apart from the nature microorganisms. So to live in our house or living environment, important point is we need to rely on the mechanical ventilation systems. So in our group, we are focusing on the especially transmission of airborne bacteria between house and community places and open air environments. And also such kind of microbe transmission is very different depending on urban area and rural area. And we in our living environment, our living environment is filled with human-associated bacteria and also bacteria from the outside. And also some bacteria living in indoor environments. So especially in our living element, one of the most important source of bioaerosol is come from bathroom. So first part of my talk, I will focusing on bathroom microorganisms. So one of the most important characteristics of Japanese style bathroom is our bathroom is separated from a toilet. So this is a traditional, conventional Japanese style shower room. Shower is like this. It means we can open the shower head like this. So it means that we can take a sample by swab inside of shower head. So using shower water and inside shower head sample, we firstly compared the microbe in the shower water and shower head microbe from all over the Japan. Protocol is like this. Just after extracting DNA, the bacterial community is analyzed using the amplicon of 16S with MinION sequencer. And also, after that step, we are focusing on the pathogenic bacteria inside the bacterial community using the pathogen databases. So this is the result of bacteria in shower head, inside shower head. So as you see, the majority of the bacteria is a Microbacterium, which is a very, very normal bacteria found in water environments. And following bacteria genus is a Sphingomonas and Brevibacterium. In Sphingomonas is also very famous bacteria and not so strange, but inside shower water, we found the Brevibacterium inside the shower head. It's not so common in water environment because it's usually a human as commensal bacteria. But in the case of water itself, the majority of the bacteria is usually belonging to Sphingomonas, Microbacterium, gravidizomium, and no human-associated bacteria are found in the water samples. So it means that shower head, inside shower head, some bacteria from human sources are growing. So this is a beta diversity. So water means blue dot cluster, and shower head community indicates that these red colors. So as you see, the how clustered the bacterial community is, water is like this, clustered very condensedly. So it means that bacterial community inside water is very similar, but in the case of shower head bacterial community is very different each other. So if when we focusing on the only pathogen in the bacterial community, this is a result of this is a list of pathogenic bacteria found in the water and shower head samples. As you see, as you know, in Japan, we can drink every tap water everywhere without any exception all over Japan. So the bacterial community is very similar, and also from the DNA sequence analysis, we found no no pathogenic bacteria with high virulence are found. For example, we couldn't find any genus Legionella. And also important difference between European and American tap water, we found only a few mycobacteria, for example. So in the case of EU and USA, the Mycobacterium genus, the percentage of bacteria is around 40% in US, and in the case of Europe, around 6 to 7%. but we found mycobacteria in Japan only average is 0.4%. It means that in the case of genus Mycobacterium, Japan Mycobacterium frequency is around more than 50 times difference in the abundance, relative abundance. So that's why now we are trying to understand if our tap water drinking system originally don't include this kind of mycobacterium, for example, don't include or not by checking the water plant system, water distribution system itself. This is ongoing work. And also, if we focusing on the pathogenic bacteria and also diversity of bacterial community, this is a plot of alpha diversity of the sample, and vertical line indicates the quantity and diversity of pathogenic microorganisms. So if we focusing on the water, this water pathogenic community or bacterial community diversity is very clustered like this. But in the case of showerhead biofilm like this, the bacterial showerhead inside biofilm cluster shows like this, totally different from the cluster of water. So it means that if we focusing on like cluster plot like this, there are so many human-associated bacteria in the sample. So if we plot like this, we found the status of bacterial community for biofilm that is totally different from water. And also, as you know, in the case of biofilm, that have to be affected by the environments, such as temperature and humidity and like that. So we try to find the what kind of environmental factor affect to the biofilm community. Now we are doing the experimental room experiments. That is the ongoing work. So now we designed experimental house in our university like this. We rent 3 different rooms in our university guest house. The difference in the room with human exist or absence, and with or without biotope. Biotope is like this, and as you see, human with biotope and only human and no human as a control. So I hope to clarify what kind of environmental factor cause human pathogen in showerhead biofilms. So, and the second topic is around the Japanese traditional house microbe. So Japanese traditional house, maybe as you see back side, this is a Japanese traditional house in Kyoto area. This kind of house is not so common, but still there are some people living in this kind of traditional Japanese house in Japanese rural area. Inside of this house is like this. This is the earth floor, soil in the house, and also normal place is from the mat of plants, and also roof is plants, and there is so many moss in the roof. So it appears very nature. So, but no one knows what kind of microbe in these houses. So we take a sample from this kind of house using swab or bioaerosol using this kind of cyclone sample, bioaerosol sampler, and try to monitor the temperature and humidity as much as possible. So from the temperature and humidity environment of this kind of thatched roof house, the temperature, surface temperature in summer is 60, winter 0 centigrade, and inside summer centigrade, winter 10 centigrade. So as you see, that this kind of temperature and humidity curve, there are some big waves. So it means even inside and outside of this house, not like a modern house, there are very big difference between daytime and nighttime temperature. And of course, it appears very nature, surface humidity sometimes reach to 100, and inside around 90% at max, maximum. And the interesting point in the this kind of very details and the continuous monitoring of humidity and temperature, we found minimum relative humidity is highly correlated with DNA concentration in from a swab sample. As you see, if we focusing on minimum relative humidity, the DNA concentration is highly correlated with this value. This value is unbelievably very high value, but it's this kind of monitoring, temperature and humidity monitoring is not so normal for normal microbiologists. But in my case, we have a collaboration with researchers from architecture. Then they calculated this value is the most high correlation between DNA concentration and other physical chemical parameters. So if we, if no collaboration with such researchers, I think we usually use only average relative humidity. Then correlation is not so high, but not so high compared to this unbelievable value. And also, interesting point of the bacterial community in this traditional house, there are so many halococcus. 70% of bacteria no archaea is microbial community is from halococcus in soil floor. Because the soil floor is made from nigari. Nigari is beaten, so it means salt is concentrated in the soil floor. So that's why halococcus is enriched in the soil. But the important point is this soil floor is made around 70 years ago. So even after 70 years, we found the major DNAs from archaea DNA. And also the water in the environment is 70% of the water community is candidatus nitrosotera. So even this environment and water are made from rare microbe. So it means Japanese traditional house is a source of rare microbe, and I think it's like a treasure.
So, 5 minutes, please. More.
Okay. Yeah, just a summary and not so many slides so far. So as you see the result, the microbe in our Japanese traditional house is totally different from modern house. And also we need to analyze more sample. So that purpose, we developed this kind of suitcase laboratory. We developed this kind of portable laboratory. And this, if we use this kind of suitcase, we can analyze bacteria and fungi community composition in 24 hours. So final slide before 2 slides. We use this kind of real-time bacterial airborne microbe monitoring system. We obtained this kind of result. So this is a real-time monitoring of viable airborne bacteria. As you see, if closed and open and closed environment is totally different in the airborne microbe. Some studies says no difference between if we open and close the window, but actually there is a big difference. So we need to find what kind of factor is important to live in healthy in our living environment is a still open question. So finally, I'd like to show you my international collaboration with, for example, Chile and Spain and so on. So if you are interested in some researches, I'm happy to collaboration with you here. Thank you for your attention.
Thank you. Thank you very much for a great talk. We have a few questions, but I will open first for the audience. Questions? This one here.
Sir, it's a nice presentation. Sir, actually you show a strong correlation with the humidity. My question is it statistically significant, or without statistics, can we depict the result?
Yeah. Yes, of course, this is statistically significant. So, but important point now is number of sample is not so enough. So we now try to increase the number of sampling point, and also we also try to compare the DNA concentration in winter seasons also, because we obtain the DNA sample only summer season so far.
There is one. Okay, there's one question in the slide. Japan is surrounded by seawater and on all sides. What are potential bacteria treat human that may migrate to indoor environment from either seawater or land?
Yeah, we already do, for example, as you see here, previously we already reported the locality of bioaerosol. So even outside in Japan, a very near area, for example, maybe you can see this mountain, and around under here is some city. The distance is only 30 kilometers, but there is a big difference between mountain area and city area. In outside bioaerosol is very different each other. So it means that even small island, Japan is very small, but still in our indoor environment is highly affected by around surrounding environments.
Okay, we have a question from Scott. Scott is online.
Yeah, so my question is quite simple. I know you're a microbiologist. I'm wondering, when you said there was no Legionella detected, did you actually culture that on charcoal media, or was that you didn't detect it by DNA?
Yeah, so that's a good point. And now, because as you know, most of bacteria is unculturable. So, and also what I detected is candidatus bacteria. It means no one cultivated yet, but I hope to try isolate the bacteria to know that its characteristics. So that's why now I'm collaborating with some researchers with high skill of cultivation with new techniques. So I'm happy to cultivate if possible, not so yet.
Okay.
Okay.
I have a question. Yep. So, Fumito-san, thank you very much for good talk. My question is along the line of what Scott was asking. He asked culturable, but I'm asking, live and dead microbes. Are you having success in, differentiating live and dead microbes using your technique?
Yeah, so yes, yes, I know you are always using PMA methods, and so unfortunately, in our cases, we just using the DNA-based methods so far, but we already obtained the RNA for metatranscriptome analysis for the samples, and also now we are doing cultivating some samples, and we have so many different kind of colonies, but not yet to determine the genome sequences. But yes, but when as in you see the I obtained the sample from the earth floor. Sometimes earth floor is made 230 years ago. Even such old sample shows a similar DNA composition. So I believe that the DNA microbe is also still alive. I believe so. I hope to obtain the result for that.
Okay. Thank you again, Fumito, for this nice talk. And let's move forward to our next speaker, and will be Onofre.
Thank you so much.
Dr. Silvano Onofre will be also virtual. Yes. Yeah, okay, great. Dr. Silvano Onofre is a professor of botany at Tuschia University in Viterbo, Italy. He gained his doctoral degree in biology from the University of Rome and serves currently as the head of the microbiological program within the Italian National Program Planetary Research, PNRA. The Space Mycology Lab, led by Dr. Silvano Onofre, studies eukaryotic extremophilic microorganisms and their survival under real space and planetary conditions, and searches for biosignatures in future space exploration. Please, Honorio, be our guest. The stage and it's yours. Okay. Can you hear?
Yes, I can hear. Thank you very much, Mr. Chair, and thank you, Venkat and Kartik, for the kind invitation. I don't know if I must share my screen, or you can show my presentation.
Your presentation is here? Yeah, okay, so they can share. Yes, can share his presentation. It's better if you do it.
Okay.
Yeah?
Okay, I share my screen.
Yes.
I did it, but I cannot see my presentation.
Yeah, we can see.
You can see my presentation?
Oh, no, nothing.
Nothing.
So, no.
But I sent my presentation a couple of days before.
Can you I think they will do that. Gotcha. Just a minute, you know.
Yeah. Now?
Yeah, just a minute. Venkat is connecting here the laptop with your presentation.
I am very sorry.
That's okay. Yeah, they'll be sharing soon. I had the same problem a few weeks ago, so I sympathize with you because it's sometimes there's a lot of technology sometimes make us a little bit crazy. And yeah, so well, it's the good and the bad of these online virtual meetings that we have nowadays. But I mean, it's good because we can be connected.
Have you my presentation? Because I sent it by mail.
No, yeah, I think they are
on Saturday.
They are, I think, uploading now and be shared soon.
I can try to change the browser, but it takes some minutes.
I think they almost there. Yes, almost. Yes, almost there. No worries, Silvano.
Okay.
Yes.
Oh,
no,
yes. Thank you very much.
Can you put in presentation mode? Thank you.
Okay, here we are. I am very sorry for the delay, but I would like to talk about my space adventure with my best, one of my best friends, that is Cryomyces antarcticus. This is the adventure of a particular microbiome that is really a community that is living within the rocks in Antarctica. Can I go ahead, or you can change the slide, please? We can start in Antarctica. Antarctica, as you know, is one of the worst environments on Earth, and namely, I would like to talk to you about the McMurdo Dry Valleys. Please go ahead. Okay, the adventure started many years ago with my friend and teacher Imre Friedman, who discovered the presence of living organisms within the Antarctic rocks. This is a very, very particular environment that I can describe, please. Oh, this is the results of the biological weathering, because it is amazing to understand that the microorganism can live within the rocks. In this case, is a particular rocks that is named sandstone. And the action of the growing inside the rocks results in this particular kind of landscape that you can see here in these slides, please. This is a comparison between the Dry Valleys landscape and the Mars landscape from Curiosity rover. And as you can see, the two environments are very, very similar and very dry, very cold, and almost without life. But also, if during the exploration of the Dry Valleys for preparing the Viking mission by NASA, they were not able to find microorganisms in the soils, but Imre demonstrated that, please. That where the environment is too harsh to be colonized by microorganisms, the microorganisms themselves could colonize the porosity of the rocks. Since this kind of rocks, please, are colonized inside. This is a section of the rocks, and as you can see at the millimeter scale, there is the presence of some microorganisms that produce some layers that are the layers that are describing the community. There is a reddish crust that is due to the fact that the iron is moved upstairs, up by evaporation. There is a black layer with fungi, black fungi and lichenized fungi. There is a white layer. The white layer is characterized by the presence of fungi that are in symbiosis with algae, and it is a lichen. And below there is a green layer with green algae that are not in symbiosis with fungi. And after that, there is this accumulation zone with iron accumulated by the leaching of the small quantity of water that is available only when some snow can accumulate on the surface of the rock, and after that the sun is melting the snow, and the snow can penetrate, the water can penetrate, giving the opportunity of metabolic activity of this very small community that is composed of only microorganisms. This is a microbiome that is, in fact, a community with producers and with consumers and so on. Please. We studied this kind of communities by the mycological point of view, and two new species has been published many years ago, Cryomyces antarcticus and Cryomyces minteri. These two black fungi, they are named black fungi because they are black, because the presence of a thick layer of melanized wall, but they are also named black ears because they are more or less unicellular with very small hyphae and filamentous behavior. Please. Oh, this is the environment in which is growing Cryomyces antarcticus, and as you can see, a microscope view of the fungus. This fungus is very resistant. It's resistant to desiccation, to radiation, to extreme temperatures, and also, as demonstrated on the International Space Station, in space as Mars conditions. Please. Okay, we know two species of Cryomyces. Cryomyces antarcticus is the most important and most resistant species, but also Cryomyces minteri. You can see picture that are representing these two species. Please. Okay, and after discovering this very interesting species, we decided to test this fungi not only in simulated experiments on ground, but also in space on the International Space Station. Please. we decided to test this fungi after some experiments that demonstrated that this mainly Cryomyces antarcticus is a polyextremotolerant fungus that is able to resist to high saline concentration, to very high temperatures, to very low temperatures, and also, and it is very, very important, it is a fungus that is able to resist to very long periods of dehydration. And as you know, dehydration is one of the most stressant conditions for organisms. Please. Okay, some papers has been published about the resistance of this fungus that is able to survive also to very, very high doses of radiation. For instance, you can see that Cryomyces is also able to resist to 55 kilograms of radiation. In this case, is irradiation by heavy ions that is very, very difficult to resist. And the fungus demonstrated to be able to resist to the, no, I was wrong. The resistance is 1000 grays for heavy ions, but up to 55 kilograms under gamma radiation. Please go ahead. And okay, and this also the demonstration that that Cryomyces is able to resist to irradiation of gamma radiation and gamma rays and heavy ions, but is something similar, a different experiment, but the results are similar. Go ahead, please. We asked, we submitted to ESA some proposal for exploring the resistance of this fungus to space conditions, and thus asked for the first proposal was to test on the International Space Station in low Earth orbit, but we are also proposing experiments in lunar orbit, the Deep Space Gateway, and if possible in the future experiment on the moon, and after that exploring Mars. Please. Oh, the LIPS program has been launched in 2001 by ESA. Please. Okay, in the framework of the LIPS program, it was possible to propose experiments on the International Space Station. Please. And this is the first step of our very long program of testing this fungi in experiment in space, but also in Mars environment. Please. The first, we had the opportunity of performing 3 different experiments. The first was the life experiments, the lichen fungi experiment that started in 2004. After that, the biomax project that started in 2009. And after that, the biosign project that possibly will be launched on the International Space Station in the near future. Go ahead. Lichen fungi experiment has been performed with the collaboration of University of Tuscia, ISA, DLR, INTA, Spain, University of Düsseldorf, and JPL, and we collaborated with Venkat for performing this experiment. The scientific questions were, could Antarctic microfungi survive in outer space? In outer space, could Antarctic microfungi survive on Mars? And we tested also the possibility to have some information about the lithopaspermia, that is the possibility of transferring life from different planets by means of meteorites. Please. This is the SPOSE subsystem that has been used for exposing this microorganism in space, please. Okay, SPOSE mechanical subsystem that has been transferred to the International Space Station for exposing this microorganism in the outer part of the International Space Station. It means in the space environment, but also in Martian environment, simulated space. It means same radiation on Mars, more or less. It means simulated atmosphere in particular holes and so on. Please. Okay, the launch by the space shuttle in 2008, please. This is the European Laboratory Columbus that is active on the International Space Station, please. And this is the radiation environment at the altitude in which is flying the International Space Station. Please. Again, the Columbus Laboratory and the astronauts from US that are working on the experiment outside the International Space Station. Please. In 2009, coming back from the International Space Station, using again the Space Shuttle Discovery, please. And in ESTEC, ISA, in the Netherlands, with Gerda Ornek, that was the founder of the European astrobiology, and we are in our hands really very emotional, the samples returning from space. Please. The first results, the simplest result is, are these microorganisms still living after being for 18 months in space? The answer is very easy. Yes, they are still living, and the demonstration is that they are able to grow. And this is the most simple experiment that we can do, but it is really reliable. They are able to grow. They resisted. Please. Okay, some of the results. Go. Very important that they were able to resist also in simulated Martian environment in space. Go ahead. And in this case, this experiment was considered by Nature one of the important experiments in 2015. daylights.
Sorry, you have a few minutes more, 3 minutes.
Only 2 minutes because I started at 11:08.
More, so just go and so we will open for the questions.
Okay, I can go ahead very quickly, but it was necessary to have 8 minutes before starting. Okay, this is the biomax experiment. Go ahead. This experiment was based. Please go. This experiment was based mainly of another scientific question. Could biomolecules that are produced by microorganisms resist in space environment, and how we can find biomolecules in Martian environment exploring Mars? Go ahead. Okay, go. This is the design of the experiment for exploring Mars. Go ahead. Go. We used the Soyuz for performing this experiment, but now it's quite impossible. Go ahead. Go. This is a short video, but go. Please go. Okay, the results was very, very interesting. Go. Yes, go. Again, go. The cells were perfectly preserved. Go ahead. Go. Okay, go. And with this experiment, we realized in a very large group a sort of database of molecules that could be searched during Mars exploration, during Mars 2020 exploration, and also Mars samples in the future. Ahead. Yes, go. Go. Very different kind of analysis. Go ahead. And this was the most important papers published on Science Advances. This is a list of biosignature that could be searched during Mars exploration. Go ahead, please. Oh, this is Biosign in the future. Go ahead. But I think that I can go to the conclusion because we demonstrated during many years of researches that microfungi that are eukaryotes are able to resist in space conditions, but also in Mars conditions simulated in space, that many biosignatures, for instance, melanin or also DNA, could be used for searching eventual remnants of these microorganisms growing on Mars. And thus we will wait the next step of space exploration for deepening our knowledge of the possibility of existence in the past or possibly also in the present of these microorganisms in other planets, namely on Mars. Thank you very much for your attention. And you can go ahead with the presentation to go to but that is more or less finished. Thank you very much.
Thank you so much, Silvano. Great, thank you. That for you. I think it was really good and quite inspiring presentation. despite the internet problems and issues, but it was great. And we have actually several questions in this Slido. And I think there is any, oh, yes, question from the audience.
Okay, I can.
Yeah, just a minute, he'll ask.
Yes.
Okay, so I will go first with the, well, there is one question in the Slido. It's what is the evolutionary advantage of being crypto-analytic?
Oh, the advantage is that in that environment it's impossible to grow on the surface of the rock. And we studied the distribution of different rock community in Antarctica, and we demonstrated that there is a border, and this border that is, of course, close to the Antarctic plateau, is the border of the growth organism on the surface of the rock. After that, it's only possible to survive within the rock, within the rocks. And thus, if you would like to live in that place, you must go inside the rocks to protect from dehydration, from very cold, and so on. This is the advantage. They are able to colonize places that are not possible to be colonized by different organisms.
Okay, great. Super. Yeah.
So, hi, this is Ishan from IIT Delhi. A question, maybe you alluded to a bit in your bioscience part. Can you take a look at a rock from the outside and tell whether it's going to be colonized inside?
But rock from outside, are you meaning meteorites?
Yeah, I mean, if you go to Antarctica now, you must have enough rock samples.
Yes.
Okay.
Yes, we collected a lot of rocks from Antarctica. You have a collection. We have also a section of a museum in Italy with these rocks that are colonized by organisms, and we know different types of communities colonizing rocks. And we are using this microbiome as a model system. Just because the microorganisms that are living inside these rocks are able to survive in conditions that are very similar to conditions that we can find on Mars. Also, if there is a big difference, because on Mars we have a very low atmospheric pressure. I don't know if...
Silvano, the question is, you have a rock without doing anything. Can you predict from the top, saying you have life inside, from the top, without doing any analysis?
Sorry, it is hard to understand.
Just by looking at the rock structure from the top, can you say that life is inside?
But I showed the layers of the community.
Because you are cutting and then doing the transect, you're cutting that, then you will be seeing it. Without cutting, without doing anything from the top, can you do any kind of, spectroscopy to see whether life is inside?
Yes. It is necessary to cut the rocks, because from the surface we are not able to observe the community.
Okay, thank you.
Okay, thank you.
Yes, thank you again, Silvano. Now let's move to the next speaker. I think the next speaker is quite special guest. I think it's myself. So.
Give me that. Already we introduced him. He's going to talk about he's in the desert, but let me see whether he's doing it at desert. Yeah. He's going to talk about the desert microbiome. Extreme microbiomes, no, that's true. Extreme microbiomes from Antarctica and Saudi Arabia.
Hi, yes, please. Thank you. Well, thank you again for Kartik and Venkat and all the organizers for this opportunity to present at least some of work that are being developing now in my new university, KAUST. KAUST is King Abdullah University of Science and Technology in Saudi Arabia. I'm actually from Brazil, but I spent the last few years. It's not my fault. I didn't know. Can you help me here? Someone? So I don't know what happened. No, it's, I can see here, but, oh, now it's okay. I don't know. I think it was the contact. Okay. Okay. I can go just the slides. No problem. Yeah. No worries. Yeah. No worries. Thank you. Yeah. So this is my lab in Saudi Arabia in the Laboratory of Microbial Ecogenomics and Biotechnology. So it's a new lab. So I set up my lab now 2 years now. So I started to organize the lab during the pandemics. So the first year was quite tough, trying to buy everything, try to build a lab from the scratch. But now it's 95% operational. So I think it's quite good. So I will show some of the work that we've been doing now. But my lab and my main interests are very diverse. So I go through my background actually is soil microbiology, soil microbiome, but I started working on extreme environments 15 years ago when I went to Antarctica for the first time to collect samples. So I've been working on Antarctica samples for the last 15 years. And now in the last 2 years, I started to work on desert and different extreme environments from Saudi Arabia. And I just would like to introduce you a brief history about the research that I developed in Antarctica. And that actually came with one question that I was asked for that during a job interview at PNNL. And Janet asked me, what is the best or one scientific achievement are you most proud of? And this was difficult because, our work and our microbes are like babies. We like our stud, our work. And so it was quite tough. But I think this is what I like the most, because it's work that went through basic science. So we did a lot of basic science, trying to know more about the microbial diversity in this case in the peninsula, Antarctica. So it's a different region. It's not the dry valleys, or it's an Antarctic peninsula. And I have a deep interest in microbes working for good to help us to cope with environmental problems, issues. And one of my main interests, it's actually bioremediation. So I was studying the microbial diversity in Antarctica, in that region, and also the potential for degradation of oil, diesel, and the material that they use to power the heat in the stations. And there was a fire in the Brazilian Antarctic Station in 2012. This was burned to the ground. And this is what I like. Our basic research collected during 10 years of research was the foundation for a big bioremediation project that cleaned up that region. And now in the last picture is the new Brazilian station that was built after the cleanup of this mess that happened after the fire. So I like this, I think, the most, because it's a full history of the basic science with applied aspects. So I like both things. Just a quick break. And Venkat, please control my time. I'm when I start to talk about those microbes, I just forget about time. So this is some previous work. We published the first there. It's not working the pointer, so sorry about that, but I can try here. Yeah, this one here is the first viral sequencing data in terrestrial Antarctica sample. And we published this in 2010, this journal. It was a collaboration with DMTH in the US. And from that time, the things changed completely. And we start to work with the next generation. So we start working on next generation sequencing. Illumina, basically this is a data from Illumina. And one thing that we must consider when we think about this kind of soil and when we intend to use bioremediation is that it's a very poor soil. There is no nitrogen at all in the soil. So you have nitrogen when you go to the penguin nests, but when you look into this, the overall soil, it's almost like a regolith. So it's really poor. There is no nitrogen, as you can see here, but we came with this gradient of hydrocarbons from the diesel spill. So it's here is a highly contaminated samples to the less contaminated sample, but there was no non-contaminated sample at all. And here is data from that gradient using metabarcode 16S. We can see here that this is the highly contaminated soil, the less contaminated soil. We can see the abundance of Cytophaga, Methylovescitales, Polaromonas, and Williamsia. Williamsia, Polaromonas. I will ask you to pay attention. I will, give the snapshot about the Polaromonas, because we have a very interesting history about the Polaromonas. So this is metabarcode, but we also perform shotgun metagenomics. And here we try to look into the functional genes that were expressed or the potentially function of those, the microbial community. And the notation of the metagenomic data, this is a shotgun, so this is not metabarcoding anymore, revealed the most abundant hydrocarbon pathway in this particular soil was related to the CYP4050 enzyme family. So this was interesting. You can see here in this red part. And looking into more details into this degradation pathway, the shotgun metagenomic data, the functional potential data, the dominant degradation pathway was this alkyl derivative PHI, polydroxyaromatic degradation, mainly methyl naphthalenes that belong to the CYP4550 family. And this was quite interesting because it was never described, this kind of metabolic pathway in such Antarctic samples. There are some other metabolic pathways for degradation of TPH and PHA hydrocarbons, but this was not very common. So we were quite surprised to see that this was the prevalent, the dominant metabolic pathway for degradation of this hydrocarbon. And remember that there was no nitrogen in that soil. And this is confirmed here. I mean, look into the NIF genes, the nitrogen cycle. We can see that the heavily contaminated hydrocarbon, heavily contaminated samples contain abundance of NIF genes. So nitrogen fixation is interesting, and of course makes sense. It's an important process when you have excess of carbon and no nitrogen, so they must have a balance. And there was a kind of selection of nitrogen fixing bacteria. And when we go to the less contaminated soil samples, we have other enzymes or of genes belonging to other steps of the nitrogen cycle. And trying to reconstruct what was going on here, we can see that the hydrocarbon degrading bacteria are also able to fix nitrogen. So this is also interesting when we think about potential new ways to apply bioremediation in different soils. And the other steps of the nitrogen cycle happening in the other samples. We decided to perform a microcosm or mesocosm, actually, because this was in situ in field in Antarctica. So using the contaminated soil and I'm being very briefly here. Okay, so it's a long story, but trying to make short. We tested several different bioremediation conditions or treatments. Remember, there was no nitrogen, so we include some fertilizers, nitrogen chemical fertilizer, in a proportion to have a balance to improve the degradation. And here is metabolic coding of the mesocosms now, the microcosms. And I, again, ask you to pay attention, especially in this particular treatment here. There's when we add some different concentrations of fertilizer, but these red bars here are the Polaromonas. Like, again, it's a beta proteobacteria. There's a new nomenclature now, taxonomic nomenclature, but I will stay in this old-fashioned because we know the names. So it's really something that is happy to be here, to be there in that sample, with high contamination of oil and in the presence of nitrogen. This is MEG, so we were not able to isolate the beta proteobacteria, Polaromonas-like microbe, but we reconstruct the genome based on the metagenomic data. So this is the MEG of a Polaromonas-like, so it's showing that there is really something there, Polaromonas. And it's just for curiosity, there is only one strain or one species of Polaromonas that was sequenced by Department of Energy in the US, Polaromonas naphthalenovorans. The name is complex, you know, but so there was not many information in the database, and but we can see that our MEG, our Polaromonas-like metagenome, genome assembly, contains several genes related to naphthalene degradation. So do that pathway that I showed before. So it's, well, intriguing. And the metagenomic data, yeah, so the Polaromonas, so abundant there, but we also perform metaproteomics. And again, metaproteomics showed us that they are not only there, but they are really working. So those Polaromonas here are the proteins, or the metaproteomic data, showing that the Polaromonas is not only there, but is also performing the degradation. And it's, again, interesting because we didn't have an idea about this when we tried to isolate the microbes there. So the metagenomics really gave some new insights. I just saw that. Even more, we performed some experiments. Remember, Antarctica is a very cold environment. Even the peninsula Antarctica is also very cold. So you can have winter time like -30, -20, sometimes even -40. This is in the peninsula. In the continental Antarctica, it's even worse. In the summertime, we have 0, -5, -10. Sometimes you can have 1 or 2 above 0 Celsius, you know, so but it's still pretty cold. And when you try to perform bioremediation in this kind of environment, in this type of environment, we must rely on this short window of the winter time, or the summertime. But here, performing some freezing towering experiments and, well, several measures, we noticed that there are some beta proteobacteria again, so perhaps maybe the Polaromonas or some group similar to that one, and some actinomycetes that are actually active even at -20 degrees Celsius. So this can also improve the ways that we can perform bioremediation even during sub-zero conditions. And I was quite happy when we published this data because my colleague Charles Greer from Canada, he also showing metabolic activity at -20 degrees Celsius in Arctic samples. Yeah, and that metagenomic paper that the data that I just presented was also a collaboration with, I must say the names, my colleague Janet Jensen and from the US, PNNL, and Lucy Seldin from Brazil. We also did some work. This is Junior, my former PhD student. Now she's a postdoc in my lab in Saudi Arabia. And so Junior was interested in thermophilic communities from volcanoes. And in her thesis, Antarctic volcano. Antarctica has 4 active volcanoes, and this is one of them, Deception Island. Actually is an active volcano. It's beautiful there. And our interest and Junior's interest was actually to study the diversity of thermophilic microbes in Antarctica, and also to perform screening for biotech applications. She just published 2 papers this year showing the potential for several isolates. Basically, Anoxibacillus was the dominant genus, and several species, and some other species as well, dominance of bacillus, thermophilic microbes, or thermotolerant, some of them, to degrade oil and produce biosurfactants. And yeah, so there is a huge potential for biotech applications. We built a huge collection, not huge, but I mean, important collection of psychrophilic and thermophilic microbes from Antarctica. That is now also, I'm also adding isolates from Saudi Arabia in that collection. So it's growing, and we are exploring the potential. And we have several novel proteases, UV resistant bacteria, very interesting data on antimicrobial resistant genes in Pseudomonas like from Antarctica, we just published this year. So it's a very interesting collection to explore. Of course, novel genus and species. And just one example, just catching and using one isolate for the collection, Microbacterium sp. So we published the genome of this bacteria, and we showed, and we could see in the genome that there was some potentially novel carotenoids, this Microbacterium. This was not known at that time. Next, we purified the carotenoid, and we did some tests with culture cells to see the phototoxicity, the toxicity of the carotenoid as well. And it was pretty good data. We published a second paper. And finally, the carotenoid was totally purified with the association with people from the chemistry group. And they prepare some emulsions of these to use as a sunscreen. And now this is in a process of patent and dealing with a big pharma company, because the industry is really interested in this. So yeah, so some papers published in this. Okay, and let's move to a more warm place. So Antarctica is, I love Antarctica, but Saudi Arabia was a kind of surprise, because usually we believe that Saudi Arabia is a huge desert, but actually it's not a huge desert. Actually, they have several different kind of deserts. So you have rock deserts, sandy deserts, mountains, deserts, but you also have geothermal sites. So there are more than 20 geothermal hot springs in Saudi Arabia, and totally, or just a very, very few publications. So it's a really open field for research. So there's a lot of things to be discovered there. Volcanoes. I had no idea that Saudi Arabia has 2,000 volcanoes before I moved to coast. Most of them are inactive volcanoes, but there are a lot of volcanoes that are still considered active volcanoes. So it's a very interesting place to start. Thermophilic microbes as well. So those are the Harats, those are volcanoes. Not only deserts, here are the volcanoes, the black and white mountains, geothermal sites, and more volcanoes. And just to show some of the work that we've been developing there, there is a amazing place. It's called Alula. It's a heritage site of Saudi Arabia. It's beautiful. It's the same civilization that built, Petra in Jordan. So they have also very interesting places there in Alula. So we decided to collect samples in Alula, and they also producing, of course, a lot of carotenoids. And we have now more than 1,000 isolates. Most of them, several of them, are resistant to UV-C, and we are studying these UV-resistant microbes now in our lab, bacteria in our lab, also targeting the carotenoids, but also some collaborations with other colleagues. Oh, thank you. So also some, the sampling in Alula. So we have a lot of data on the microbial diversity, those bacteria. And now we are trying to isolate and purify and characterize the carotenoids. And we also have the genomes of most of those microbes, not totally. I mean, we're still, it's still working in progress, but we have very interesting data about some novel, potentially a novel genus and species of bacteria there. And we are also trying to search for new CRISPR system in thermophilic bacteria. It's also quite interesting. Just brief, the carotenoids or the pigments, you can have several different pigments and they have a huge potential, not only for the cosmetic industry, but also for antitumoral properties, antimicrobial properties, and several other applications, including sunscreen that Venkat mentioned in his presentation. And we decided to play a little bit because those microbes not only interested for biotechnology perspective, but of course, astrobiology perspective. And we decided to test those microbes using Mars simulants. It's commercially available simulants. And using metabolomics to try to identify which metabolites are produced by those pigmented bacteria under UV radiation in a Mars-like soil. So this is a commercially available simulant. So yeah, there's survival and growth, and another thing, we also add perchlorate on these Mars simulants. And the idea here is not only here is of course astrobiology. Perchlorate is abundant in Mars, but the idea with this kind of study is not only astrobiology, but we are also testing these microbes and how, if they can improve soil quality somehow, that we can also apply here on Earth, you know, in environmental biotech process, that we can increase soil quality as well and try to increase or the possibility to have crop plantations in a arid environment like Saudi Arabia or any other desert region. So this is a brief, the pigmented bacteria, perchlorate. And this is a work that we have been testing now. This is Al Haba crater. It's amazing volcanic crater in Saudi Arabia. There is a huge level of salt in the center of the crater. And we isolated some very interesting black yeasts there. One, we know that is exomophila, but the others are completely different. I think new things, that new species, or I don't know, we are still now working on the genomes of this the fungi. This is interesting. Mars, the first one here, yes, and the other is Saudi Arabia. Mars, Saudi Arabia. Of course, it's not cold, but they have snow during the winter in this region. This is Tabuk region. This is another surprise for me. I didn't know that they could have snow in Saudi Arabia, but it's in the north part of Saudi Arabia, in that particular desert, in the Tabuk region, they have snow now in December and January. So it's quite beautiful. So we decided to build a Mars simulator chamber. And this is a small device to play with our isolates. And for that, we had a collaboration of people from Blue Marble to give some insights about the way. This is still a work in progress. I mean, we have the project now, but we still try to, elaborate the best possibility. We are working on desert biocrust to improve soil quality. So it's another project that it's open to collaboration. So this is why I want to showcase this here. New metabolic pathways. We isolate very interesting thermophilic bacteria, autotrophic actinomycete, that actually is producing those very symmetric carboxysomes and is fixing carbon using two different pathways. Both are active because we have proteomic data, and now we are trying to collect metabolomic data. And maybe maybe 3 carbon fixation pathways. So this is quite new, was never described or reported before. Start a collaboration with Venkat and try to use the supercomputer that we have at CAUS. Just to make a link with the next section tomorrow, we are also trying to build some organoids because we are trying to use models to study microbes and the interactions between those microbes. And my crazy mind is thinking about use those organoids and challenge with some of our microbes, the good microbes, and use our Mars chamber simulator next year. Let's see if this will work. But those are the images or the figures of the first organoids built in Saudi Arabia. This is my student, and I have a collaboration with people that are using with 2D cells, and there was no one doing many organs in Saudi Arabia. So we decided to start this and to push this field forward. Beautiful landscapes in Saudi Arabia. So it's not only the terrestrial environments, but also the Red Sea can have a very deep and extreme environments. And we are also working now describing for the first time the first hydrothermal vent in the Red Sea. So this is, we are finishing the microbiome work, and this paper is on the line, finished, yeah. Main goal of microbiome initiative for tomorrow. And for the students here, please take a look in our paper that we published this year in Nature Microbiology. It's a perspective. So we gathered several different experts in microbiome science from different fields, completely different fields, and we try to put some words to leverage the power of the microbiome to protect biodiversity. So it's quite interesting. I recommend. And that's it. And it's the best momentum that we are living, especially for the young people here, to be working on microbiome science, because it's really, really, we have the tools that we didn't have in the past. So it's the best momentum now to study microbiome science, and they can help us in agriculture production and even help to conquer the space, as the first section was showing. This is my group in Saudi Arabia. So it's a very diverse group. I have Saudis, but I have also blue hair. I have different people working there. And so, yes, thank you again.
Thank you very much. And audience have one question.
I have two questions. One is about that in the beginning when you mentioned that in those Antarctic soils, which were devoid of nitrogen, you discovered that they're nitrogen fixing. But where did they take the nitrogen from? From the air?
Yes.
Oh, wow.
Yeah.
Amazing. Yeah. And the other question was one about that they were decomposing hydrocarbons, but were they producing water? There was H2O plus CO2. They were, you didn't mention it. So they were releasing water and CO2.
Yeah, exactly. Yeah, I didn't present the data hydrocarbon degradation, but we have this data as well. So it's a complete process to the end, not only by one microbe, but when you have the microbial community, they are really processing the degradation of LTPH.
So they were metabolizing.
Yeah.
Water is metabolizing.
Yeah,
Okay.
and this nitrogen fixing bacteria that is also able to degrade hydrocarbons. This is a quite interesting approach, because I published a paper in 2002 about this, and there was no many, I mean, you know, people said, oh, interesting, but that's it. But now it's coming back with really strong power. So there are several groups trying to use nitrogen fixing bacteria, so that maybe you can, you know, if you combine this, if you have a SYNCO, a synthetic microbial community, you can try to minimize the use of fertilizer. So there's some combinations and some help of the engineering.
But it's actually also important that some microbes, they make their own water. They don't need water.
Oh, no, this is in the decomposition process in the end. Yeah, but yeah, degrading the hydrocarbons. So the final product will be water. But they are not making water for themselves, you know. Yeah, yeah, but there'll be a water in the end.
And my last question is about the middle part of your talk, when you said that they're resistant to UVC. But this is amazing, because UVC is completely blocked by ozone layer. UVC does not pass through. Why would they be resistant to UVC?
Why not? Because we even go down to the deep sea, 3,000 meters down, we find the bacteria that is resistant to UVC. So you have a genetic cassette that actually help different kind of radiation resistance. So whenever you expose to UVC, that kicks up and then help it. But that may be using.
But they never exposed to UVC. They can't be exposed to UVC.
Doesn't matter. That's the beauty of the microbe. Whenever it needs, it kicks its genetic thing. Until then, it is silent.
Yes, exactly. You can go to the middle of nowhere in Antarctica or in a desert, and you find some microbes resistant to several antibiotics that we have. Even the
Never been
yes, exactly.
He's having a question.
So I had one question. Like you mentioned that you had isolated around 1,500 microbes. So like how exactly did you isolate? Like which media and which condition did you use to?
Several combinations. Now the collection is more than 1,000.
Yes, sometime during the break time.
Yes, sure.
Try to find him, because it's not a simple answer. But maybe the one that Karthik put it there. Do we need newer and better annotation tools, metabolomic data to handle the exotic microbes being sampled in all these extreme environments?
Yes, perfect.
Answer that first question. That question.
Oh, there are more questions there. Okay. Do you need newer and better annotation tools? Yeah, sure. Definitely. Yes. Metabolomics is still in I think metabolomics is the next frontier, actually. I mean, we should improve metaproteomics, metagenomics, of course, the pipelines, the tools, the bioinformatic tools. But metabolomics, I think it's quite interesting, because it's the way that they communicate to each other, the microbes, and also the way that they communicate with the host. So we need to understand a little bit more about this. And I didn't talk about this, but we isolated some very interesting metabolites from those black yeasts and some siderophores that were also never described for this kind of fungi before. Yeah,
Thank you. So we are not going to stop, 6 minutes over. You can start now. Here, and
so thank you so much. Our next guest is known as PB. It's Punyalok Baduri. And Dr. Baduri is a professor of biological sciences in IBSE, Kolkata, and he has established and currently leads the Center for Climate and Environmental Studies. It's an interdisciplinary center on Earth and environmental sciences with societal relevance in Kolkata. He has published more than 90 peer-reviewed papers in international journals. His research interests include studying biological complexity systems, biologically complex systems from coastal and deep sea environments using taxonomic and molecular tools, and the role of environmental factors, including nutrients, in shaping the structure and function of biological systems from these environments. He is the recipient of the prestigious Swarna Jayanti Fellowship Award in Earth and Environmental Sciences conferred by the government of India in 2019. So Yep, preparing the presentation. Just a minute or two. Yeah. Okay. Yeah. Yes, problem solved.
Okay, great. Thank you so much, Alexander. Sorry about the glitch, but I think now it's under control, it seems. I'm going to take over from where Alexander stopped. Yeah, he showed one of those slides where microbiomes and mangrove environment, if I'm not wrong. And I'm going to end up this story for today with my talk, where I'm going to highlight why microbial biocomplexity is very important to decode. And if you want to understand critical processes such as ecosystem functioning or biogeochemical cycling, you need to address the biological complexity at microbial scales. The importance of ocean in shaping our civilization is not unknown. Last 5,000 years, ocean has played a very important role in influencing our civilization, and this has formed the basis of a concept in the 21st century, the blue economy concept. Except I would use the word sustainable blue economy, which is very important. We are at the we are celebrating the UN Decade of Oceans, So it's very pertinent to understand the ocean from a land boundary system. So it's a land-ocean boundary system. The coastal ocean and blue economy is an important component. If you look at it, 1 in 10 livelihoods globally are dependent on the fisheries. And that actually underpins about food sources of almost 3 billion people globally. So how important ocean can be. And it has been estimated that the potential value of the ocean economy will reach almost $5 trillion by 2030. In Asia, the importance of coastal ocean driven blue economy is very significant. It can be as low as 1% to as high as 87%. So therefore, the health of the ocean is very critical. And if we want to understand the health of the ocean and want to make it sustainable, we need to look at it from the biological perspective. Microbes provide a very important picture. Now, one other thing we need to keep in mind is there are cultural and educational values, which we cannot be measured, from the perspective of ocean. So that does not really fall under the blue economy as a whole. Now, when you talk about biological complexity in ocean and coastal ocean, essentially what you're talking about is the functional relationship between this complexity in biological communities and, for example, biogeochemical cycling that is happening in the coastal ocean. And that actually plays a very important role because the physical chemical complexity of the coastal ocean is extremely dynamic, and that seems to shape many of the microbial guilds that we kind of try to estimate or try to measure in coastal ocean. And one of such manifestations of this complexity is by looking at the plasticity of the genome or microbial plasticity. Now, how do we address this plasticity, or how do we look at this biocomplexity? One easy approach, relatively easy approach, I would say, is looking at time series. So imagine a scenario, we are in this auditorium, let's say at 8 a.m. in the morning, there was nobody, at 6 p.m. there is nobody, but in between there is so much activity has happened. And imagine 1 year down the line, you're able to figure out 31st of October 2022, how many people are there, who are there, where from, all the information is written. And that's what we call it as snapshot, essentially. And time series is nothing but an ecological snapshot that covers 3 important verticals. One vertical is the physical vertical, that is the physical measurements of the ocean. Second one is the chemical measurements of the ocean. And third one is the biological measurements of the oceans. And once you start to measure these verticals, what you end up is with some extensive temporal data set that can help us to understand how environmental drivers can shape biological complexity. And you can look at the scales of microbes, for example. Now, globally, there are different time series out there. Of course, many of them are very famous. You know, you've got the Lobo time series, the Hawaii time series is there. In the Atlantic, you've got the Western Channel Observatory. In the European seas, there are many of the regional seas-based time series. For example, the Helgoland time series. In the Indian Ocean region, there is only one time series, the Rama time series. So essentially, if you look at a basin scale, the number of time series are more in the Pacific Ocean, in the Atlantic Ocean compared to the Indian Ocean. And time series that looks at particular types of biotopes, you know, those kind of time series are very limited at a global scale. Now, why do we take the pain of looking at all these things? Why do we have a time series, as I was saying? Well, if we look at this picture, this is a microbial loop in coastal ocean, a paper that was published by Alexander Warden a couple of years back. But here, this is a very simplified system. Of course, as you go towards the land boundary, this system becomes more complex. Why? Because you've got freshwater flow. That means there is a lot of terrestrial runoff is happening. Organic matter is coming from the land. And then you should not completely rule out the interaction between the land, ocean, freshwater, and the atmosphere that is happening every microsecond. And then there are some vertical pillars are there from the biological perspective. You know, for example, bacterioplankton, you know, all bacterioplankton are bacteria, but all bacteria are not bacterioplankton. You know, you've got cyanobacteria, we say, but there is something called marine planktonic cyanobacteria. They're very different. Their entire life cycle is planktonic phase. You've got diatoms that form associations with bacterioplankton. So they will survive only when there is bacterioplankton is there. And then these pillars then actually starts to interact with processes, for example, the carbon dynamics in the mangroves, as an example, or can drive more bigger scale processes like the benthic pelagic coupling. So therefore, time series can play a very, very important role. And this is a paper published in 2020 by Allard et al. Alexander was leading this work, where the research priorities from a microbial angle for mangroves was highlighted. So mangrove is a very interesting system. It has evolved, or it has developed, this biotype has developed at the land ocean boundary system. And some of the mangroves, they actually receive a lot of fresh water on a daily basis. And I'm going to talk about that very quickly today. The Sundarbans mangrove, this is the world's largest contiguous mangrove spread across India and Bangladesh. The area is almost 10,000 square kilometer. It's a UNESCO World Heritage Site. It's a Ramsar site. And I wanted to set these mangroves on a realistic context. You know, you look at the freshwater flow, 42,000 cubic meter per second to about 120,000 cubic meter per second. So that much amount of freshwater is coming from the 3 major rivers, the Ganga, Brahmaputra, Meghna, and they are dumping a lot of this organic matter from the land, from the vegetation, ultimately into the coastal ocean. So the net essence of that is you've got suspended particulate matter load, which is between 200 to 800 milligram per liter, probably the second highest in the world after the Mississippi Missouri. Okay. And therefore, light penetration is extremely poor. Nevertheless, there is plenty of aquatic primary production that is going on. And that's the enigma in the system. And the system as a whole is, of course, very well known. So this is how, like any other mangroves, the Sundarban mangroves looks like. There are plenty of endemic species are there. And of course, this is famous for the swamp tiger. And I would argue the healthy microbiome of the swamp tiger is there because of the healthy mangrove microbiome. And therefore, the maintenance and the health of the mangrove is very critical. So what we did is way back in 2010, in the westernmost part of the Sundarbans, this red blocked area, we set up a time series program. So imagine now you can measure all these three verticals: physical, chemical, biological, vertical. And here I'm showing you a simple plot of how the oscillations in salinity has changed over the years. So I can go back in 2010 and figure out what was the salinity like, and versus, let's say, what the salinity was in monsoon of 2020 versus the salinity in 2012. So those kind of questions we can address. So this sampling actually happens on a weekly basis or a bi-monthly basis. Now the other one that we have is a seasonal time series. So as you go from west here towards the east, so this is the dense part of the mangroves, protected, very high biological diversity. Here we have seasonal sampling. We have got 250 stations that we sample out here. And of course, this activity is happening mostly either in the pre-monsoon time, that is the summer, or in the post-monsoon time. Now, I wanted to highlight there is something very interesting about this time series side, the seasonal series. Look out here, there is a dip in the salinity, and that is happening in the pre-monsoon time. It should be the reverse. Salinity should increase because, the air temperature is very high, surface evaporation is happening. The reason why that is happening is there is groundwater seepage is happening in this area. So as a result, the salinity is becoming more diluted. So you could have microbiome that is from the groundwater mixing with the saline water of this time series site. So here is one example. What we have done here is slightly different approach. We have actually taken a traditional approach and integrated with the third generation sequencing. So we have done V3, V4 using MySec platform Illumina. And in parallel, we have done clone library of the entire 16S RNA molecule. Okay. Now, what is interesting is when you analyze the 16S RNA molecule from the clone library, you see that in the monsoon or post-monsoon, there are different types of communities are showing. And you can see different ecotypes of Synechococcus are showing. But when you look at the V3, V4 region of the Illumina, you hardly see those sequences being represented. So entire molecule gives a much bigger perspective than a region of it, particularly when you're looking at marine planktonic cyanobacteria. And what we found out was that although we thought that this system is completely driven by diatoms, that is not the case. Marine planktonic cyanobacteria can account for almost 10% of this algal production in post-monsoon season, and we have calculated that value between 0.4 to 14.7 grams of carbon per meter square per year. So that's a huge thing, taking into account the area that we are talking about. What we did is we took those sequences, what we got from our libraries, clone libraries from the Illumina sequencing, and also we looked through the available database of sequences from different other ecosystems where the nitrogen conditions are very similar. Okay, so South China Sea, Baltic Sea, Monterey Bay, and these are all coastal sites. So we started with 2,487 16S RNA sequences. From there, we narrowed down to 438 sequences, which are truly marine planktonic cyanobacterial sequences. And when we did the deep phylogeny of it, we found that there are a couple of clades out there which are very interesting. In particular, one clade, clade 8, was found exclusive to Sundarbans, salinity tolerant. Not surprising because remember, there's so much freshwater flow, and of course, there's semi-diurnal tidal pattern. But we also found there's one clade where this is present in Sundarbans and also in South China Sea. They lack the nitrodeductase subunit B, and some clades are found in Sundarbans and Baltic Sea. So this ubiquitous nature of the microbes we found, but it could be that depending upon the niche, the functions can be very, very different. So it's one thing finding the sequences, and the second thing, a completely different thing, trying to prove that actually the organism is there. So we are quite lucky. we're able to establish a culture of organism from that cladate. And we named this new species Synechococcus murigangai. And we found that this organism can grow along a range of nitrogen sources, very high concentration of nitrate. And it has got a very complex pathway, where you can utilize urea. So you think of a scenario, the system is getting urea. Of course, it can come from agricultural land. Why run the entire nitrogenous machinery? You get the urea, you actually use that and assimilate the nitrogen ultimately into amino acids. So this system is very active, and not to our surprise, when we sequence the genome of it, we found that there are a lot of genes, which, for example, the FDHA, which is linked to DMSO superfamily, so essentially potential role in aerosol formation, iron binding proteins, urea metabolism pathway, and to our surprise, we found the entire protein machinery that is present in flagella and pilus formation. So we do not know whether in our culture we did not find the presence of the flagella and pilus, but we anticipate, we think there are some experiments that we are still doing to show that probably these organisms are able to steer vertically into the water column because the light penetration is very low. Only in certain times of the year, the light actually penetrates into the deeper layer. So therefore, this apparatus can be very, very important. Just to give an example, we found in cyanobacteria diatoms also, we see the same thing. We found a new species of diatom, Thalassiosera sundarbana. We actually described this diatom very in depth. We have done medpair PRN library, entire genome sequencing. We have drafted the genome, it's about 150 mbp approximately. We are right now doing the PacBio to understand the genome. But what is very interesting is you can see from this image, there are some organic threads are coming out. And in these organic threads, there are bacteria attached. If you remove the bacteria, the diatom will not grow. But if you take this bacteria out, they will grow. So the diatoms are dependent on the bacteria for the growth. And the reason for that could be that these bacteria can play a very important role in preventing the diatoms from getting, fouling organisms or biofilm being formed on the frustule. So that's something which is very important that we found. Now, why do we care about the microbiome? Because essentially, when you look at the microbiome, you understand the temporal or spatial variation. We can understand what it means from the perspective of the coastal fisheries and what it would mean for the fisherfolk community. So there is a intrinsic relationship with the biological communities and the livelihood. I quickly go, I remember I showed you this seasonal time series site out in the eastern part of the Sundarbans. And there are the signatures of the freshwater, microbial communities that are showing their presence during the summertime. And that is because of this flow of the groundwater seepage into the surface water of the Sundarbans. The last but not the least, we see that in Sundarbans, there are some unique signals of microbial communities are there. And these are mostly talking again from the water column. We compare it with many other estuaries of the world, and we found that there are some unique groups within the Firmicutes and Bacteroidetes that might be very important in modulating the carbon cycling. And we have shown that the breakdown of the mangrove litter is being done by very specialist group of bacterioplankton that are being found only in the Sundarban system, again, using Illumina sequencing approach. Now, this ecosystem or these kind of ecosystems faces multifaceted threats, I'll very quickly go. One of the threats that we face is, anthropogenic nitrogen load. The nitrogen load is reaching very high. That means it reaches a condition, it becomes unidirectional, will end up with heterotrophic bacterial bloom. The second thing is, we are seeing a lot of these ARGs and MRGs in the Sundarbans water. This we have done using the grid ion sequencing, and we are running an environmental surveillance program. We are going upstream of the Sundarbans, looking in the river Ganga to identify the AMR hotspots. So we know that where from these AMRs are originating and where it is ending up. The other big problem is microplastic pollution in mangroves in Sundarbans. And here you can see an Indo-Pacific finless porpoise died being entangled in microplastic. This is the horseshoe crab, which is more primitive than the dinosaurs, a living fossil. And we find different types of microplastic fragments. We know the chemical compositions of those. And interestingly, those microplastics actually acts as a plastisphere, and the biological communities, the microbiome changes. Not only that, these can become the ground or the hotspots for AMRs to persist. So you will end up with transposon probably being shifted, and wanted to highlight that this plastisphere can become a substratum for microalgal colonization. Here you can see a diatom actually has colonized the microplastic surface. So that has led to the establishment of something called a mobile sequencing laboratory monitoring mangrove health in a changing climate. This is being funded by the Bell Foundation along with the EDNA Collaborative. We are working, and our idea is to mimic this or to reciprocate this across many of the mangroves in South Asia, so that we can do a healthy environmental monitoring. Now, anything is incomplete because we are talking of a huge area. So sensor development is one area where we are working on. We are developing cost-effective sensors to track the changes that are happening in the environmental gradients in coastal ocean, including in mangroves. And the idea is to understand how the health of the oceans are changing. So why do we need to all of this? Well, we need to do all of this because we want to address the SDG goals. We also want to have a sustainable blue economy, but we can only address these SDG goals by sharing and innovating. And this symposium is a fantastic example where we are sharing our thoughts together. And how do we innovate it? A classic example we are doing is a concept called geobiomatrix concept, where we are restoring the mangroves in Sundarbans by actually adding bacteria, adding microbial formulations that can do hitchhiking and mimicking and help the mangroves to survive. You can look at the poster of Amrita Maiti. She's from our research group. She's doing a PhD, and look at it, how she's addressing this question. And where do we want to end up? We want to end up in a scenario, ocean has to be sustainable if we want to have a healthy life and a healthy lifestyle. We need to take a very pragmatic approach, and microbiome plays a very, very important role. We need to understand the microbiome, and we need to use the microbiome to make our ocean, make our ecosystems healthy. I thank our research group who are doing all this hard work, all the funding agencies, and of course, the organizers of the 4th IBSE International Symposium. Thank you so much.
Thank you very much. It was a very inspiring presentation. And we have time for questions in the audience.
Very, very nice presentation and a very good thing. So you said this, I don't know whether I heard wrong. All the time you were telling fresh water, fresh water. So what is the salinity of the Sundarbans water?
Maybe around 10, 12.
10, 12, okay. But it is estuarine. It is not a fully fresh water, right?
Well, in summertime,
one third of the salinity, yeah.
Summertime it becomes zero.
Summertime.
Yeah, and monsoon time it becomes zero also.
Okay, so now you might have done seasonal variation, over the period of a year, where the Ganges running a lot of things in will have a different population compared to the, high saline time. So what are the things that you are seeing most different?
Well, I mean, this kind of system from a bacterium perspective, you get a lot of Vibrio. You get a lot of environmental Vibrio out here. But increasingly we are seeing, we have, described a lot of this Vibrio also. I did not show out here, but we are increasingly seeing that many of them has, very extensive pathogenic gene islands. So my fear is, I think what can happen is if these AMR hotspots continue to release these AMRs or the AMRs originate from these areas, these are ultimately ending up in that big estuarine system, and it's not completely estuarine. Remember, 2 times or 3 times in a year it becomes zero. That's the time, things can go, beyond control, because most of these bacteria, they can grow in zero salinity also. And therefore, these transposons or the mobile genetic elements, through these genes can get transferred, and you could end up with a scenario with a bacterium which can be potentially very dangerous.
So one other question about the symbiotic bacteria in that algal stuff that you have shown. So you have done the metagenome to figure out what that particular bacteria. You said you cannot grow them without either of them, right?
Yeah, those are Oceanimonas.
Oceanimonas.
Oceanimonas genus.
Oceanimonas.
Yeah.
So you have the You don't have the culture, but you have the sequence, so you have the mag.
No, we have the culture, we have
Ah.
the sequence also.
So it can grow in the absence of,
The bacteria can grow in the absence of the diatom, but the diatom cannot grow in the absence of the bacterium.
then you have got the diatom not growing without, okay.
Yeah, the diatom you have been cultured only when you put it in Oceanemonas, it grows. If you remove the Oceanemonas with antibiotic treatment, then the diatom doesn't grow.
Is it superficial or inside?
No, it's actually connected to the chitin threads.
Thank you.
Yeah, thank you so much.
Well, there's a question there. Towards the end, you mentioned the new plants and microbes are being introduced for revival in the mangroves. Of mangroves. What kind of microbes are they? Actually, it's also one of my questions. You can talk later, please.
Yeah. So most of these microbes are a combination of photosynthetic, oxygenic photoautotrophs, plus you've got heterotrophs. You've got actinomycetes, you've got heterotrophs like Oceanemonas. And the beauty of this, what you're doing is these bacteria and these cyanobacteria, they can form, ECM, extracellular matrix. And the ECM, what it does is when you apply that, the ECM actually brings in stability of the geobiological layer. And essentially the sapling will not be taken away by the strong current. So that's what we are applying to do that. Yeah. Yeah.
Thank you, Alex. And
So just one thing, you are invited to join the Mangrove Microbiome Initiative, okay? Don't forget this.
okay.
Thank you so
So
much.
thank you, Alex, and give a big applause for today's speaker.
Thank you, guys. Thank you. Thank you, all the
And
speakers.
there are two things. That's right. There are two things. One, you have to give that. That's what I'm trying to say. So one is, we are requesting this session speaker to come here. We want to give some memento there, right? That's one. The other one is, after that, please come. May not be there. Okay.
Tomorrow, just before the coffee break, you said it's going to so we can have it just at the start of tomorrow's first coffee break.
Okay, we want to have a group photo for tomorrow. So that's a second thing that I wanted to tell you guys. So first,
one more thing is, I think on the schedule it's printed that Thomas's talk is at 8:30. It's actually at 9 o'clock.
Tomorrow morning, right? Yeah, 9 to 9:30. I should give it to them.
I request Dr. Kasturi Venkateshwaran to present the memento to Dr. Rosado. I request Dr. Rosado to present the memento to Dr. Baduri. We will break for the poster session while enjoying the coffee. We will be back at 6 p.m. again at the ICSR auditorium for the documentary film on amazing caves.